Alrighty. Let me double check and make sure everything is working on the live. I think we are live. Welcome to Growing with Fishes, episode 147. This week we have Kevin McKernan. Thanks a lot for joining us, Kevin.
Yeah, thank you for having me. I appreciate the time to share some info with you guys.
Yeah, it's really awesome to finally have you on. But a lot of people recommend you, and you have a very knowledgeable person. You've done a lot of really amazing research. Thanks for taking the time to join us and educate everybody on some of the different things that are going on right now in the industry.
Yeah, you bet. So what should we start on? What's the topic du jour these days?
So why don't we start off with giving everybody a little bit of background about yourself and a little bit about what you do.
Sure. So my background, I got involved in genetics very early on in 1996. I got lucky enough to grab a job on the Human Genome Project back in Cambridge at MIT, and began working on the R&D team there. And we built a bunch of the robotics to actually make millions and millions of E. coli clones to sequence the human genome. And around 2000, when that finished, I spun out a company called Agincourt, which became a very large DNA sequencing service provider. And so I'm very intimately involved with data policy and data release policies that may have encumbered some people in the cannabis space. But one of the themes we had on the Human Genome Project was to put data publicly as quickly as possible, within 24 hours if you could. That was kind of the agreed upon Bermuda principles for the project. And if you're contributing to it, put your data public as quickly as possible. Otherwise, other people are going to waste resources sequencing the same thing. And it was good policy. And as a policy, we tried to also encourage in the cannabis field. But before I got into the cannabis field, this company, Agincourt, began building a DNA sequencer called the solid sequencer. Excuse me. And this sequencer ended up as a spin-out company from Agincourt when Agincourt got acquired by Beckman in 2005 or 6 it was, 2005. We became the largest sequencing provider in the country by then, and Beckman Coulter came and picked the company up for the DNA purification tools that we had built and also the sequencing lab that we built. But we spun out this 19-person company that a year later Applied Biosystems came and bought. And it built a sequencer that had the promise and eventually achieved a 100,000-fold reduction in sequencing in a matter of like 3 or 4 years. So an ABI was the big giant in the field at the time. They owned the whole sequencing market, and they bought this company to compete with a new startup called Illumina that was starting to clean their clock with a new DNA sequencer. So I played that race for 5 years, building faster and faster versions of that sequencer, and even got involved in acquiring some other IP and platforms known as Ion Torrent, which was a semiconductor sequencer, which does things very differently, but still very, very interesting. And then around 2011, I started getting the itch to leave as the thing had been maturing and been in that fight for a long time. And that's when I had a lot of friends that were asking me about cancer, mainly because the sequencers we built, the first place people put them was on sequencing tumors. And if you sequence someone's genome and you sequence their tumor, you can find differences that might be druggable specifically in a tumor. So this is a big personalized medicine, excitement that was happening. The only problem was there weren't very good drugs to drug them with. While we could figure out what mutation was in the tumor, you usually were picking through a wardrobe of like taxotere and spitin, which are all toxins. So a few of my friends who had cancer were asking, what about cannabinoids? And I knew I had a very young relationship with cannabis in 7th grade till all the way through college and even after, but never really in the light of cancer. I always thought of it as a bit more of a recreational drug and something that shrinks tumors. And I went down that rabbit hole, and anyone who's been down that rabbit hole knows how deep it is. And that made me want to just quit my job and go sequence the cannabis genome to figure this thing out, because I was somewhat shocked that no one had done it in 2011. This was many years after we had sequenced maize and after we had sequenced Rapidopsis and a host of other plants, but cannabis was like on the wasn't even on the list. So I started a company called Medicinal Genomics in 2011, and the first thing we did was sequence that genome and put it public in a couple, 60 days. We started the project in July. We put the data public in August. And it was a ChemDog line that wasn't spelled correctly, which was a huge embarrassment. But that's just what we were given. You know, it was spelled. Oh, I think I may be seeing some lag here. Are you there? Can you still hear me? I may have got a freeze here. You know what I'll do is maybe I'll kill the video so you have better bandwidth. That'll work. I'm losing your. I can't hear your sound there either.
There you go. That was on my end. My internet connection died for 2 seconds, so if there was a brief cutout,
Okay.
that was my fault. I apologize.
All right. Well, I've left you with my my fancy mugshot there instead to save bandwidth. But so anyway, no one had sequenced the cannabis genome yet. We started medicinal genomics in 2011. We had to set up a BV actually over in Holland to do this back then, but that we put public in 60 days just to figure out what do we have. And it was a horrible reference at the time, but it was the best the technology could do in 2011. I think John Page picked up some of that data, published Finola, and compared it to the ChemDog sequence we put public, and also compared it to Purple Kush. But that kind of got my feet wet in the cannabis field back in 2011. And fast forward now, and we finally have the technology to read the genome correctly. We didn't have it back then in 2011. The plant genomes are much harder to sequence than human genomes or human tumors, for that matter. And so only recently have the technologies evolved to the point where they can manage reading the cannabis genome. And just this last August, we kind of repeated that same story again and sequenced a type 2 plant this time, known as Jamaican lion, and put that public in 60 days. And that was a really fun project because that project we funded with a cryptocurrency. We got this cryptocurrency Dash to basically fund the sequencing of the genome, and that's a very interesting currency. That's it's a branch of Bitcoin, but has different governance in it that can actually hand out grants. And we applied for one, and were awarded one in June. Used it to sequence Jamaican lion and put it public in August. And the reason we picked that strain was severalfold. I think we have a few folks joining. Let's give them a chance to introduce themselves. Well, I think you've got my introduction. I won't go on to the Jamaican lion story until we hear from Tara Lee and a few others. That's my background. I do a lot of genomics, and I'm only recently in cannabis.
Thanks for joining us, Tara. Well, yeah, I guess Tara is having connection issues too, so I guess everyone's having problems tonight. So I got a really good question for you because you're one of the few people that could answer this question. I do apologize for the reconnecting and disconnecting of the other app. We're trying to dual stream to Twitch, and we're having some side issues on that, but at least the YouTube's working just fine. So cannabis, I've heard that cannabis can actually take in genes from nearby plants or terpene profiles from nearby plants. Is that true, or is that just a myth?
Well, that's an interesting thing. So I have not seen these studies done on cannabis. I have seen other plants where they use antibiotic fumigation and then measure the terpene profile before and after applying antibiotics, and they completely change. So without a doubt, the terpenes are in fact quite sensitive to the environmental conditions. That's one reason why I don't think they're a very good fingerprint for patents. You know, we've all seen this biotech LLC patent come out, and that was one of the reasons we were sequencing the Jamaican lion genome. Is there some evidence of that thing predating the patent and matching the patent claims? I'm not a patent attorney, but we took a stab. I'm familiar with the patent system. I've used it before, and I just took a stab at a strain that looked like it predated it, and we'll see where that ends up in court. But basically, they have in that claim a non-myrcene dominant type 2 plant. Anything with a BT-BD allele and a ratio of like 1.4 of THC to CBD, or CBD, I'm actually, the ratio may have backwards there, but it's a ratio of CBD to THC that makes it a type 2 plant. And it has to have a BT-BD allele, which is a type 2 plant. So the claim on that patent has genetics and chemotypes mentioned. Probably important to have both, but if they had patents on just the chemotype, I'd be concerned, because I think the smaller the carbon molecule gets in cannabis, the more direct line it is with the environment. So when you change the environmental conditions, the first thing that's going to change in the plant are the first steps in the synthesis pathway of IPP and GPP. And these are the kind of the Legos, the 6 carbon Legos that begin to build terpenes. But you're also competing with those in making cannabinoids, and the monoterpenes in particular. So, but the cannabinoids are like 3 or 4 enzyme steps later. And so when you change the environment, there's a whiplash effect that has a fair amount of time before it actually hits the cannabinoids and tweaks those. So the first thing you're going to see change are the molecules in the front end of the pathway, which are all the small, short molecules involved in terpene synthesis. So you antibiotic fumigate these things, microbes are going to start to change. The first communication that's going to go on is going to be small molecule. You're going to start to see small molecule changes. That's why I think it's a little bit difficult to have patent claims that are only on the chemotypes, because those aren't necessarily reflective of the plant, they're reflective of the terroir. Now, I think there is some truth to the fact that some plants have knockouts and just will never make certain terpenes. They're genetically programmed that they can only perhaps be a limonene dominant, but you may see that limonene is no longer dominant under a different condition, that its second or third terpene may predominate and take over as the major terpene if you shift the environmental conditions. And so there's a combination of genetics and environment there that I think make the claim space on the chemotypes a bit too broad and too vague. And too many people can accidentally fall in them with actually completely novel genetics in the plant that's been bred to be a completely different varietal.
That's really interesting, yeah, because I've heard a bunch of different people, and I've heard people even talk on that, and I know I've repeated it a few times about that. I know everyone's heard the whole strawberry kush from Kyle Cushman and everything like that. That's why I wanted to ask a geneticist if there was any, truth to that.
Well, there's evidence in the literature with Arabidopsis and other plants that when you wound them, they start emitting small molecules like ethylene and other compounds that alarm the other plants around them that an herbivore is nearby and turn your immune system on. So there is certainly communication that's been documented, particularly in the pyrethrin pathways. If you look at the pyrethrin pathways, which are still questionable as to whether there's some extinct versions of them in cannabis that we found. We put out a preprint about that, but we don't have the mass spec data to really prove it's exclusively making pyrethrin. It could be making another molecule that has the same molecular weight. But nevertheless, pyrethrin has been studied in chrysanthemum, and when you start wounding those plants, they emit these molecules that turn that pathway on, which is an immune response pathway. And so they tend to do things like grow those things at very high altitude and under stressful circumstances, much like you'd see in cannabis, to raise the pyrethrin production. So there's no doubt, you might get some bizarre artifacts if you grow plants right next to your trim room, because you're probably outgassing tons of small molecule gases the plant puts out during trimming that may signals the plants next door that something's coming to eat us pretty soon.
That's really good, really interesting. And now, will you get different expression from, say, pruning the roots versus pruning the leaves, or is that? I don't know.
That's a really good question. Yeah, we have seen studies that when you apply some of the precursors to the cannabinoid pathway in other plants, when you apply them to the roots, they trigger immune systems. So this is one way to fight botrytis, as there's been some publications of people applying some of these compounds to the roots. And the interesting thing is that the carbon-labeled compounds, the carbon labels demonstrate they don't get through the roots. They just trigger something in the roots to fire the immune system up and fight off botrytis. So I believe trimming roots is a whole other story I hadn't even considered.
So you've done a lot of work with cannabis genomic mapping and things like that. So what is possible, and there's a lot of different controversies around this kind of topic right now, especially with the phylo stuff. What are the things that you can actually learn from the genetic code, when someone sends a sample, and what are things that people can't learn from the sample? Okay, so that's a very timely question. So there's been a lot of, I think there's been a lot of miscommunication, particularly on their end. When they're challenged with this question, they tend to reply with the Jurassic Park straw man. We can't Jurassic Park your strain, and that's a misdirect. There's a lot you can do with the genetic information, which I'll touch on, but I'll even just to back up a little further.
they were receiving leaf smears which have cells on them. And how alive or dead those cells are is still a question mark. There's people I've spoken to in the tissue culture arena that claim they could culture those things, but it wouldn't be easy, and you couldn't do thousands of them at a time. So I think it's important we specify what in theory could be done and what there's any evidence of them doing. And there's no evidence of them having done that. Maybe they're getting accused of it. I don't know. I've not seen people accuse them of that. I've seen people say, hey, look, you took in at least 5,000 samples for the sequencing. Their CEO is on record saying they're getting 10,000 XY tests a month. And the XY test was listed in their investor document as like a massive data intake tool. So I think we have to back up and say it's probably more than 5,000. It's probably 20 or 30,000 DNA samples they've collected. I doubt they have been tissue culturing any of those. I think that's a huge leap for them to be able to pull that off. So I don't think it's fair to accuse them of anything there's no evidence for. I don't think it's fair to say it's impossible that they can't be doing it. I think that's important for people to know that if they're getting any remnant of cells in the mail, a gifted tissue culture person could potentially culture those back. Let's assume that's not happening because there isn't evidence for it. I think what's important to focus on is they have thousands and thousands of genomic DNA samples, and they're acting as if they don't, those are meaningless. Like the tests that they ran on them and put public aren't useful. I agree with them on that. The tests that they ran and put the data of the test they ran and put public is not very useful information. We've put out some PowerPoint and videos on this, not necessarily calling their data out directly, but data just like theirs, that you can't actually get their data through the standard QC tools and sequencing. It throws out most of their data due to some artifacts. So it's not a very, you might be able to get some fingerprints and some parentage from that information, but there's not enough marker density to do any type of breeding work with it. So I don't think they're being dishonest there. Where there's gray area, where we don't have information on, is that they're in front of investors saying they have a 50,000 SNP array, but they only gave you guys or whoever their customers were 2,000 SNPs when they did their sequencing. So did they sequence 50,000 and just give you guys morsels, or did they keep another 40,000 for themselves and make this chip? I don't know. It does beg those questions. Where did they fill that gap from? Was it from customer samples? They had a huge library of customer samples, so they probably used some of those customer samples to design these arrays. They have been at some scientific conferences demonstrating that they are using the Galaxy to pick out samples that they want to further study and sequence their entire genomes of. They have a few slides they presented suggesting they're going to do 99 of these genomes to high depth of coverage, and that they're specifically picking them out of the Galaxy. So when they say they're not using your data, that might be true. The data they gave you, they're not using. But the data that they have in terms of your genomic DNA, they have that on ice forever. And what you can and can't do with that today is a bit of another misdirect, because what you can and can't do with that data tomorrow is a whole nother discussion as to how quickly genomics is advancing. What you can do today is you can PCR amplify any gene out of that library, put it into an agrobacterium vector, which is a way of cloning that gene into a bacteria that likes to insert its DNA into other plants. So if you can tissue culture your own plants, not theirs, not anything you gave them, but your own plant, if you know how to tissue culture, you can make a very small amount of tissue culture, infect that with agrobacterium to move a gene of interest from another plant into your plant and have transgenics. Now, they claim they're not doing that, but that is what is doable. We have no evidence that they're doing that. But when people say it's impossible to use your genetic library to improve our breeding program, you have to be a horrible molecular geneticist to say that. It's unbelievable that they wouldn't want to leverage or advertise that library to a potential investor or an acquirer.
Well, and I was hoping too to just better understand, what is possible with the samples that people have sent in to not only Phylos, but there's probably 6 or 8 different companies out there that are mapping genomics, and some of them have ethical people running them, and some of them don't. Right. I know why don't you tell people about the company that you do? Because I know you actually have ethics, so that's one of the reasons why I wanted to bring you on and have someone that's actually not trying to turn everything over to Monsanto immediately on the show, that could actually speak from a point of science and a point of education.
Sure. So in 2016, maybe it was 2015, we caught wind of their business plan. It wasn't a confidential business deck. It's kind of like one of those Benzinga things where it probably circulated around some investor community. At least it didn't have confidential written on it, but it clearly laid out their premeditated plan to harvest people's data and then turn on them. It was from at least back in 2015. This was their plan. When we saw that and tried to voice caution to many people in the field, particularly some of the people closest to them, that, hey, this looks like an ethical violation, it wasn't met with a whole lot of warmth, let's say, and maybe my methods of warning people of this came across as insecurity, but effectively people turned to me and just said, "You're just their competitor talking shit. This go away. It's the sour grapes." And so we did go away. We stopped talking about it for a few years, and just decided the best thing we could do is build tools that would be an antidote to this type of heist. And so we began building tools that would allow growers to do this. And we began building blockchain tools to put people's data in their hands as opposed to in ours. So I have one of those here. Let me see if I can manage to turn this thing back on. If you put this video back on.
The video, that wasn't on your issue. You can turn your video back on.
Oh, all right, cool. So there we go. This is a little device. That's about $850. This, what you're seeing in there are 16 wells of a mini PCR device. It basically runs off of a USB port, and your phone connects to it with Bluetooth. It runs on iPhone and I think on Android. But what it does is it runs these little tests that are sex tests. So if it's yellow, it's a male. If it's pink, it's a female. So we can do XY testing at Grow, and not you, which means the data never leaves your facility. And that's where they're getting the lion's share of the data. Now, we can't do whole genome sequencing yet with things like this, but that's coming. Maybe if the accuracy of this thing ever gets better, you might be able to do it on these nanopores, but they're still a little rough in cannabis. We're trying to work that out. But right now, PacBio seems to be the leading edge in sequencing, and those don't fit in a backpack yet. But these devices can not only run XY testing, but we also have built assays to pick up the CBD allele. And that's the allele that tells you whether you're going to be either a type 2 or a type 3 plant. If you have one of those genes, you're going to be a type 2. If you have two of them, you're going to be a type 3. There's a second test to help sort that out, which is the THC test. So if you want to know if you're a type 1 plant, you run, that'll tell you if that one lights up. That will only light up in type 1 and type 2 plants. So we already have 3 breeding markers there people can look for with a remote device. And some breeders have already done this. Seth Crawford, probably over a year ago, got onto this and started breeding for CBG lines just by screening for plants that never lit up on the CBG test, or the CBD test. So when you look for, if you cross 2 type 3 plants, and you cross enough of them, some of them are going to be double negative CBDs. They won't make any CBD, and then you're stuck making CBG.
I don't mean to interrupt you. Can you do a real short what the different tiers are for people that maybe don't understand what you're talking about?
Oh yeah, sure. So I'm sorry. So the first cannabinoid made in the synthesis pathway is cannabigerol, and then your plant is a type 4 plant if it only makes that. But if it has an active THC synthase gene, it will convert CBGA into THCA. If it has an active CBD allele, it will make CBDA as well from cannabigerol. It'll actually fold cannabigerol into those two. So if your genes are knocked out and deleted in both THC and CBD, your plant can't move it CBDA, and you get a CBGA dominant plant. There are other plants out there where maybe those genes are weakened a little bit, and so they make a little bit of THC and CBD, and you get a bit of a background. But Seth happened to figure out how to get like a 400-to-1 CBG line using this device and also orange photonics to confirm it, because we can tell you have no CBD alleles in here. There's another hypothesis that your plant could be, is it could stop one step before making CBG and be a cannabinoid negative plant, which is known as a type 5. So he had to figure out whether it was a type 4 or type 5, and he used orange photonics to fill in that gap. But he did all that point of grow. He did not have to send any DNA outside of his facility. And that's the model that we're trying to build at Medicinal Genomics, is everything we do in the sequencing front, we want to convert into assays people can use in the field. and do that as quickly as possible. This is why we're putting all these references public. This is why we're putting all of our data public, is because when there's more public data, there's more assays that need to be built. More people figure out what assays need to be made. We've already had people reach out to us saying they found some markers we haven't found and they want us to make the assays. So we believe that that model is more attractive than trying to own the entire breeding space as one company starting from, we're 15 people. We're not going to become Monsanto and compete vertically from soil to oil. That to me seems like you're biting off an enormous piece of the marketplace. To be an expert at all of that is very, very difficult. But what we are very good at is making these tests and doing genomics. But we don't believe we need to necessarily own all that data. So what we do when people do want to sequence with us, and this is very important, I think, in light of what happened with Phylos, is we put our data, I shouldn't say our data, if the customer asks for the data to be put public, it's put public within 24 hours of being generated. And we also timestamp the publication of that on a Bitcoin or Dash blockchain, which is an immutable ledger that we can't control. And if, God forbid, someone came and shut us down, your timestamp is not tied to our survival or to our business ethics. It's tied to a globally distributed ledger that has billions of dollars of money behind it that's probably not going to accommodate your desire to edit that timestamp. And that's what I think is probably happening right now in the galaxy, is that whole system is you can edit. I don't believe anything that comes out of it because the people can change the names. The data behind it was compromised. And on top of that, they didn't manage the password security very well. I had access to their admin password, which means I could have shuffled the whole galaxy on people. I didn't do it, but if I had it, their competitor, I bet 10 other people had it too. And so I don't think you can look to that as a legal system of who owns what, because of the lack of security, the fact that it's not, you don't have immutability in it, and the fact that the sequencing behind it is fairly compromised. So unfortunately, I don't think it's going to clean up the IP space as they. I think it's actually probably created a bit more of a mess.
Thanks for that amazing breakdown. That's so cool that you're able to provide those kinds of tools for people that they can actually utilize right in their own, their own farm. I know you did a lot of work with bringing down the cost of a lot of other genomic testing. Do you want to talk for a minute on that? Because you really kind of changed the landscape, or worked with a group that really did.
Yeah, thank you. So the since we put out, we had an XY test out probably 2 years before Phylos existed. So people have been using that for a long time, but we've gone through 4 different improvements on that, mainly because the Y chromosomes we kept running into were different. And tests we designed many years ago didn't work on those lines, and we had to adjust it so that it would. And after 4 iterations of that, I think we've probably screened more complexity than anybody with the XY test. But the price of it has also gone down 300% in that time frame. And that's not from doing a tremendous amount of engineering on our end. A lot of that is just volume. When the volumes go up in genomics, the price collapses. And so as more and more people do this, you'll see the price drops occur from whether it's PCR or whether it's in the sequencing front. The sequencing front has also improved over time. We're now sequencing 4 million bases for the same price as what Phylos was sequencing. And that's 10 times or 12 times the amount of territory that they were sequencing. So I think you're going to see an update to that fairly soon that's going to sequence even more territory. So the price of access to cannabis genomes is collapsing right now that we have better references like Jamaican lion and other data sources that we can point to. The other thing that the field hasn't had, John Page did some of this work. He did great work doing RNA sequencing, but he did this RNA sequencing back on those compromised sequencers back in 2011. No fault, it's just the technology evolution that happened. We're all working with those sequencers back then. Very recently, we have tools now that can read RNA at single molecule levels, so we can figure out alternate splice sites in RNA, and that each gene in the genome doesn't make one protein. It makes an array of proteins based on how it shuffles its RNA in the process of getting expressed. And we couldn't see that very well in 2011. And now we have tools that can see that beautifully. And we've been able to do it in the brother of Jamaican lion, which is a male plant, We've been able to do it in its flowers, its leaves, its roots. So we've got 5 different tissue types in a male Jamaican lion plant and 5 different tissue types in a female Jamaican lion plant. And we've also done this in a seeded clone of Jamaican lion, so we can see what genes are being turned on in the seeding process. And that's important for food. Right? You need to know the adestin gene. You need to understand how that thing's being expressed. So we've got that data source now on top of having done the first methylation maps of the genome. So methylation is important in genetics because it's what your environment does to your genome. When your environment is acting on your genome, it's painting it like Bob Ross with methylation, right? It's just decorating it, and happy little methylation spots everywhere. And it puts them typically on genes it wants to turn off and shut up. And so you can utilize that methylation information for genes that are specifically getting turned off in the seeding process versus in the flowering process. And you can also maybe hunt down the hermaphroditic tendencies in the plant. And that's one project we're working toward, is to figure out what loci in the genome are driving the heritability of hermaphroditism, because it's clearly a heritable trait, but it also has large environmental components. And to do that, you need the genome and some of this methylation and RNA information to really understand which genes are driving that. So I think in the next couple of years, we're going to start to get more and more of these markers that give you information about the molecular clock in cannabis, like cloned your plant 30 times. Is it now still functional? Well, the methylation and RNA signals are probably going to weigh in on that. Likewise, the hermaphroditic tendencies people want to try and get out of the picture during the feminization process, we'll probably know more about that as well. But we never had these tools, and now they're there. And a lot of this has it's actually come together out of charity. I mean, after we got this DASH grant to fund the genome, everybody started piling in resources. We've had people sequencing all these other genomes for free, and just committing them to the community because they believe in the plant and they believe this is the right thing to do. It has been a scientific arena that has great medical importance and completely politically squashed, and everyone wants to reverse that. Fortunately, while there is a lot of scare about Monsanto and other big ag industries, there are other large companies coming in that don't have their history, that are doing very good things in the field. New England Biolabs is one of them. They've got a Nobel laureate there who Rich Robert discovered restriction enzymes a long time ago, and is very cool who can resources to things like this. Likewise, Pacific Biosciences and many other players have chipped in time, resources, and brain power to try and put all this stuff public. And so I just don't want to have a witch hunt for all corporations. I think it's very different. You know, there's some that are doing the right thing.
Oh, absolutely. We had a bunch of questions from chat. So the first question from chat is, how many tests can you do before you need maintenance or recalibration on that unit that you showed earlier?
That is a good question. I'm going to have to ask the manufacturer. We don't make this device. This is made out of a company in Cambridge. And I hope it's a lot because they put one in the space station. If they have a problem up there, they're going to have a real problem. But I will get some information from PCR on that. That's we haven't seen one go out of spec, and we've used them for a couple years now. But an engineer has that info that I don't have, and I will find it.
Okay, cool. We also had, so another question is, could you ask if there's a way to screen photo period trigger, short day versus long day, or autoflowers through the genomic testing?
So we don't have our eyes on the flowering loci yet. That is something I know Phylos and others have been targeting. I think for those who saw some of the threads on Instagram, they were trying to make the claim that they weren't looking for anything in your data, but the password to the galaxy was autoflower allele. So I think they were. So we haven't been necessarily hunting that down. We'd love to talk to people who have maybe a good scientific explanation for how to hunt that down. But it is an interesting loci, and that if we can find autoflowers and we can tag that and screen it in one of these portable tests, then people could do some very interesting crosses to maybe grab another harvest out of a year if they're going outdoor. So I would love to catch up with anyone who has those questions, and if they can, inform us on a direction to go, we'll share what we know and what we can do.
We had another question: is what sequencing method do you use, and what marker genes are you looking at?
Awesome question. All right. So we use a couple methods. When we do whole genome sequencing to build references, we use mostly Pacific Biosciences sequencers they have a new one out called the Sequel 2 that does 50 to 100,000 base pair reads if you can get DNA long enough. And we just did another male genome with them. They just delivered us a beautiful male genome. It's the most contiguous male genome done to date that just rolled off this week. It's a Jamaican lion male. So that's what we use to build references. Once we have good references, we use Illumina NovaSeqs to basically, you can plow through about 20 to 40 genomes on a NovaSeq and get whole genome, 30 to 50x coverage on a NovaSeq run. Illumina is very short read sequencing, and you can't use it to build a de novo reference. You can only use it to map it back to a reference you've built with PacBio. And once you have these PacBio references, you can then resort to these cheaper, higher throughput systems. We also have played around. We have our own sequencers and MySeq internally as well. HiSeq. We've been tending to batch things and send them to NovaSeqs recently, but we occasionally resort to those for doing some sequencing. Now, internally, we have two different flavors of sequencing that we offer to the public. One sequences 4 million bases in the cannabis genome, and the other one sequences the entire genome to over like 12x coverage. They both use Illumina. One is a whole genome shotgun where you just shear up the genome and sequence it. The other one is a capture system that uses something from Agilent called SureSelect, which hybridizes down 30 genes of interest in the cannabinoid and terpene pathway, and then 30,000 other markers across the genome. And that's the one that we just had a 420 sale on. I think it was going for like $200. You could get 4 million bases done. To put that in comparison, I think people were paying $285 to get 300,000 bases out of maybe 200,000 out of phylas, 2,000 markers versus 30,000. So it's cheaper than what they're doing, and it's tenfold more data. That was one of the reasons we were a bit perturbed about what they were doing in the field, is that not only were they giving me data, they were taking you for a ride in terms of the most expensive DNA sequencing test I've ever seen.
Well, I'm glad you're sharing with so much of this information, because most of us aren't educated enough. I know I was almost intimidated to bring you on the show because I don't feel like, at the beginning, I didn't feel like I was almost educated enough to ask you the right questions even on a lot of this stuff. So
hey, your folks are asking about the sequencers. They know something.
So yeah, another question is, can you please explain how much of the chemovar you can interpret from the genomic data?
Oh, so is there a way to share a screen with this platform? Let me see. I think I can see it.
You go up to the little gear in the circle there, or that square, and if you wait.
All right, there we go. So, can you guys see this at the moment?
Yes.
Okay,
yes.
So this is Cannapedia. So this is similar to a Galaxy-like system, other than it's blockchain coordinated, and it looks at more than just our own data. It tries to soak in everything in NCBI, which is a database that a lot of people, it's a pain in the ass to put data in, and it's unfair to ask people to shove it all there, but it is someplace the patent office will occasionally look. So what you're looking at here is, what strain are we looking at here? This is a USO 31 strain John McKay sent us. We did not get chemotypes from him, but assuming it's a hemp line, what you can see from the sequencing over on this graph on the left that's in red, I don't know if you can follow my mouse click through this thing, but there's 3 different regions in the genome we look at to figure out whether you're type 1, 2, 3 plant. And we can determine which one you are just from the sequencing alone. We can't tell you exactly how much cannabinoids you're going to make. I think that's a little bit, maybe in the future we'll have that, but I think there's also some environmental component to that. But Etienne de Meyer has shown that this allele pretty much predicts the ratios really well, just maybe not the magnitude. But what Etienne didn't know is where these things sat in the genome and where to look, and we've since figured that out with all the sequencing we've been doing. So when you see a lot of DNA sequence coverage over here, these red spikes over here, this tells you that you actually have sequence coverage over the gene that makes CBD synthase, and so it's probably going to make some CBD. If you don't have any green or blue sequence, that means your active THC allele doesn't exist, and your other broken version of CBD synthase doesn't exist. That usually tells us you're a type 3 plant. So we can figure out type 1, 2, or 3 from the sequence alone, and of course, who you're related to and all that other fun stuff. Now, that's a hemp line. Let's look at a type 2. A type 2 plant comes out with sequence coverage like this, where you can see there's coverage in the active allele, on the active allele of the THC gene, so you can make both THC and CBD synthase. And we've got one broken copy. So this one might be a little imbalanced. We may end up with a 2-to-1 ratio or something with a plant like this, but we know it's going to be a type 2, roughly. We can also tell how inbred it is. And this happens to be a Jamaican lion. So this is really, really, really low heterozygosity because it's most closely related to the actual reference. And then if you look at Blue Dream, you would expect this one was a Blue Dream that's in Cannapedia from looks like Qualcan sent this in, and this one has no active CBD allele, has an active THC allele and an inactive CBD allele, but this coverage is really low. So this is the type 1, and you can see that from the actual terps that they, or the cannabis they sent in. It's in fact a type 1, and that matches the genetics. Now, what we're doing, we've been sequencing over the genes that make a lot of these terpenes you see up here. Some genes make many of these, and so we're still trying to correlate. Like the gene that makes beta-caryophyllene is a certain, they call them CSPT in a number, which is a cannabinoid, cannabinoid sativa prenyltransferase or terpene synthase. There's CSTP genes and CSPT genes that you have to keep an eye on, and when you do that, you can begin to predict what some of these are going to make. Now, we have some evidence on some genotypes that are starting to point to terpinolene. Philippe Henry's done a little bit more work on this. He's got, I think, some genetics now that are starting to point to 3 or 4 other terpenes. And so I think in the next year, you're going to start to find more predictive power out of just the sequencing alone. And what that means is that you can begin culling your plants 6 weeks, maybe 10 weeks earlier, just by looking at the genetics and screening them out, saying, I want all type 3s, and I want type 3s that are maybe don't have any myrcene in them. And eventually we'll start to have genetics that, we already have the genetics that look at the cannabinoids. Now we need the genetics that look at the terpenes. There is one cannabinoid we're still working on, which are the varins. We believe there's a mutation in a pathway. We're still looking for that. We don't have that one nailed. But when we do, the plan would be not to hoard that and turn into a breeder at that time. It would be to make one of these UPCR tests that you guys can run to screen for varin genetics in the field. And so if that makes sense, there's a way that you can see it from the DNA sequencing information, but whenever we have markers that are this clear, we always make them available on one of these devices so people can actually screen for them remotely and not feel like they have to send their DNA in to get real breeding done. But I think that's an important point to highlight, because a lot of people were scared when they saw, oh, Phylos is going to breed us out of existence. I think they're years behind what you can buy off the shelf already. They're still trying to find the CBD marker. We've had the CBD marker on the market for like 2 years. And so you can get XY from us, CBD. We also have all the microbes nailed in these assays. So if you want to look for, let's say, botrytis, powdery mildew, russet mites, fusarium, we have assays for all of these, both on UPCR and on a quantitative platform where you can do some more interesting work on. So all of these assays are available to people to be screening for things that have endophytes, don't have endophytes, whether they're filtering and completely making things clean. We have tools for those. And then whether you have any of these various genetics. There's already a large slug of assays people can use. I think they're years behind on catching up on. So I don't see that as a huge threat at the moment, but they do have all that genomic DNA, and I do think that is a very valuable resource that they've kind of undersold its potential, at least to the grower community. They seem to have oversold its potential, perhaps, to the investment community.
So you've done a lot of work with different pests and pathogen testing and things like that. Can you, and this is, and I'd love for you to touch a lot more on that, but before we get to that, I'd love for you to tell me, can you actually test for things like, say, hemp mosaic virus or other viral infections that are actually, you can transfer in seed stock?
So we've made attempts at 6 different viral assays. There's a couple things that make them difficult to validate. One is most of these hemp mosaic viruses or hemp sun viruses or cryptic cannabis viruses, they're RNA viruses, which means the molecular marker for them decays pretty quickly in the field. Like in curing, you're not going to get it by screening cured material. We've had many people try to get us DNA or RNA from leaves that have images that look like they might be infected by a virus. Some of the problems with that is a lot of those images also, they're not unique to a viral infection. You can see a brown and patterned leaf, and that can be Pythium. That can be a host of things that are destroying your root zone and giving you chlorosis on your leaves. And so it's not that the image of mosaic virus, I don't think, is signature enough to know what to look for. So we've gone down the road of trying to find it in those samples, and we've not been successful yet finding it. So we either have the wrong molecular target, or we've not figured out how to sample this thing in a way where we get the RNA to be picked up. The good news about these tests here is the enzyme that is in there will amplify RNA. So we designed this with the intent that we'd eventually have an assay pop positive for that. We've just not found one yet. So that's contact us if you think you have something. We certainly want to hear and see more photographs and perhaps see if we can work with people to try and pick them up, because we have some assays in the shelf that are just rusting because we've not yet found them ever to go positive, other than you give it synthetic DNA and it does what it's supposed to, but it never seems to pick up a virus on the actual plant.
I have some seeds that actually do have a percentage that express with tobacco or hemp mosaic virus. I'm happy to send them to you or whatever I need to do.
Really?
Yeah.
All right. We should talk, because there's other things passing in seeds. We've noticed botrytis can come through, and there's a possibility of many of the other endophytes are coming through seeds as well. Penicilliums in particular are probably coming through.
Yeah, it's really interesting to see how much of the things that you can actually transfer in that actual, the seed versus the, tissue transfer or wind or, some other pathogen transfer.
Yeah, that seems to be the hardest part of tissue culture, is how to get a cutting that doesn't have endophytes, or get them all out once you put it in a culture of some sort. It's just going to grow a bunch of crap in the agar if you don't have a very good sterile technique, and you're taking very small amounts of tissue. So that's the whole game, I think, in TC, is making sure you understand the microbial loads that were present before and you've eliminated them after.
So we had a couple of other questions. What about homogeneous F1s in cannabis? Do their genomic studies reveal where we are getting one genomic set over another when combining genomic data or different cultivars?
So the most homozygous line we've seen came from Franco out of the Congo. See if I can pull that guy up here.
Rip Franco.
Now, yeah, I know, it's sad. This one cost him his life. I think he died trying to get this thing, and trying to see if I can get it up on Canopedia. That was presumably a landrace, and I think I can just go up here and search for Congo, and there we go. Let me get the share screen on again here. Entire screen sounds good. So this was done a long time ago on the earliest version of our kit, but it is related to nothing. Very, very low heterozygosity, and is very far away from everything else we have. Now, it is. Would you call this homozygous? What you have to do is plant those seeds and see how distant all the seeds come out, and you can do that with the sequencing work we do. The other thing that may mature is apomixis. If we can figure out how to make apomix in cannabis, they've done this in a couple other growers. Tim Charbel's real good at this. That will allow you to make double haploid seeds, which would be purely homozygous seeds. And if they figure that out, it probably would require a GMO event in cannabis to do it, and it would probably need to be done line to line. But if they can pull that off, then the plant starts making double haploid seeds, and everything comes out like clones. That's the holy grail in other ag industries. It's not yet, it's hard. It's not yet been played out in a lot of agricultural crops yet. It's made it in a couple experimental grasses, but that is the goal. The people trying to make that happen in cannabis so that you can make plants that make these double haploids. But it's a really important point because that's what separates clones from seeds. I mean, everyone's going to clone all day long until they can get the polymorphism rate down in cannabis so that the siblings don't come out so far apart.
And for those people that don't understand what that means, what would that mean kind of on the commercial slash, you know, backyard grower?
Well, there's an investment required to make an apomic line, and so they're only going to do that on certain lines, not on all of them, and maybe those do become the Budweiser of the field. But you know, when you look at the brewing market now, that's what I see in cannabis is that there's going to be people making Heady Topper and Boom Sauce and all of these like really exotic craft cannabis strains. That market's probably going to be much bigger. It's going to grow faster than I think the microbrew or nanobrew market will, and that's still a very sizable and attractive market. So I think they both ask for different breeding things. Like if you meet with some of the people who are growing these millions of square foot grows, they have problems no one's thinking about in craft cannabis. Like they want small buds that get through their trimmers and their machines and don't clog things up, and that's no one's doing that for bag appeal on really nice Humboldt grown strains. Right, so what they optimize for is like what fits their trimming, like shear blades. That would probably never occur to anyone in the cannabis space that we need to make plants that have really uniform tiny buds. That's something that's just an artifact of them making Bud Light. But when you start looking at making these more, I think, fantastic Heady Topper like strains, it's going to be a completely different set of requirements that you're breeding for to make that pull off.
One might say outrageous new strains.
Yes, there you go. Yeah.
Sorry, I had to. So we had another question. Are there any closer to elucidating whether PM is a systemic endophyte?
Okay, so I think this confusion comes down to people's definitions of systemic. So in the genomics field, we often consider something systemic that we can detect in the bloodstream, or let's say in the plant vasculature. And when you can pick up DNA in the bloodstream, you say it's systemic. But it may not be alive in the bloodstream. It may just be shedding its DNA into the vasculature, and you can pick it up. So in the case of PM, the way that infects is it puts a haustoria into the plant as a taproot, but then it grows a mycelium network on the outside of the plant. So the vast majority of its biology is on the outside of the plant, kind of acting like sand on top of a solar panel. It just obscures photosynthesis doing this. But it does not, as far as we know, move into the seeds and get into the vasculature and be infectious internally to the plant. But we believe you can probably pick up its DNA in the vasculature. From the work that we've done. So I think microbiologists will typically say it's not systemic because it's not growing inside the plant, it's growing predominantly outside the plant with a couple taproots. But geneticists might say it's systemic because we can pick it up through a needle biopsy in the stem. And that means the DNA is circulating, but not infectiously circulating. And I think that's the important differentiation, that if there's DNA there to be detected and it's not infectious, well, that's just DNA circulating, no big deal. Botrytis is different. Botrytis we think is actually infectious through the vasculature.
So can you tell us, you've done a lot of work with, research around pathogens and pest resistance in cannabis specifically. Do you want to talk a little bit about that? I know, especially with the talk of the PM resistance, you're kind of the guru on that, so I figured I'd ask.
Oh, well, so I wouldn't say I'm the guru. There's another gentleman, Punja, up in Canada that's done some work on this as well that was very helpful for us figuring this out too. But we sequenced a PM genome a couple years ago. just to pick up a marker. So we didn't do a very good job sequencing it. It was a metagenomic mess because the genome is really hard to sequence, and there's all these other penicillium strains co-present when we sequenced it. So we didn't get a beautiful assembly, but we got enough of its ITS sequence to make a marker out of. And the ITS is the region you use to classify the taxonomy. And when we got that, it was like 98% identical to Golovinomyces, which is another form of PM that hits wine, but it wasn't identical. So we were unwilling to give it a name. And I think Punja's actually done a little bit more work here, and he thinks he's nailed the species in Canada down to an actual genus and species. But anyway, we make markers to look for that. Now, when we make markers that pick up PM, it's with a test that really only lyses open the mycelium because the spores are really hard to lyse open with powdery mildew. They're really resilient. So when we're doing a biopsy or a paper smear of a leaf and doing PCR in that, we're predominantly picking up mycelium. If your plant has mycelium on it, it's susceptible. So it's a proxy for whether you have post-infected plants that are susceptible that you can't yet see the colonies of PM on it. And it probably has about a 4 to 5 week window where you can pick up the PM before you can see it. So it's something you might want to use to screen clones. Because when you do cloning, you stress the plant out, and sometimes the PM pops out during cloning. But we don't have the loci yet nailed for PM resistance. There's a bunch in hops that have some homology in cannabis that we're looking at very closely, that we're sequencing over when we sequence these strain seed panels. But we've not yet found a correlation with PM resistance in any sequence in those regions at the moment. So that's still an open field, and I know Phylos is going after it, and good for them. If they figure that out, that could be a very valuable marker that people want to test for. It doesn't sound like they're going to be making it available to anybody else, but they might be able to use that to breed their own PM-resistant lines. Which, as Sam and others will tell you, you can still do that without these tools. I mean, these tools might speed it up, but people have been selecting for PM-resistant lines without pipettes for a very long time. And so there are ways to do it without these tools. We just hope these tools are going to make it a little bit faster.
That's awesome. Let me thing here. One second, I find my questions here.
Thanks for joining.
Yeah, thanks again.
I really appreciate it. I'm just here to listen and learn. So.
Oh, cool. It's a pleasure meeting you. Thanks for having me on.
So what areas of research in the cannabis genomic world would you suggest are open to immense depth of research for someone maybe as a college student or someone really wanting to get in that's kind of on the entry level? I know I had one of my previous roommates recently was a biologist major specifically around genomics. What could you recommend to people that are trying to basically get your job in the future but pick up from where you left off?
Bioinformatics. I highly recommend they learn how to analyze the data because we're drowning in more data than we have eyeballs to look at. So this is a field where you need to, it teaches you how to align DNA sequences against one another and look for mutations that matter, that might be coding mutations, to be able to comb through a lot of this high throughput sequencing data. It can be very compute intensive, and so it does require a mixture of computer science background and some biology background. But those are very rare people, and they're paid very, very well. They're in high demand, and that is where the biggest bottleneck is in cannabis genomics right now, is without a doubt on the analysis end, and we need more people who can study that. I also think we're going to see different levels of acceptance of breeding to GMO in cannabis. Not that I necessarily agree with it all, but some of it's already happening. We're seeing people move these genes into yeast and engineer pathways that will probably make GMP grade single molecule marinol that maybe works, right? As we've seen people chemically synthesize this, they're going to start to get yeast to make this. And the jury's still out as to whether that's going to be more efficient than the plant. We don't know.
Well, to me, the biggest bottleneck, as someone who's even worked with some of the dab pens and some mixing some of the stuff, is when you start mixing and matching different terpenes and cannabinoids, they're extremely, unless you really understand the exact ratio that you're combining them, they're very unpredictable. Sometimes they'll emulsify, they'll solidify, they'll crystallize. They do all kinds of things. They don't behave, you know, in a easy to understand, easy to. I'm sure you can map it out, but they can be, you know, very unpredictable once they get into different ratios. At least in my experience, and you can tell me, you know, how wrong I am on that.
I don't have a whole lot of experience on on making edibles or any of the any extraction techniques, so I'm a bit aloof there. But I have done a lot of work with emulsions in the past with DNA sequencers, and emulsions seem like a tool we need to understand when we're dealing with these lipid-filled compounds mixed with terpenes, which seem to be sometimes water soluble, sometimes you know, partially miscible. In oils, you're going to get emulsions, and that's a fascinating field of chemistry. It usually means you're going to have some surfactants involved to control it, which may not be in everyone's desire in these vaporizers. But yeah, that side of the chemistry has never been. It's very, very new, and there's just as much I think excitement and promise there.
So have you done much research with the alcohol-bonded cannabinoids like CBT and some of the other ones? Because there's very little research, and very rarely hear people talk about that particular group of cannabinoids. Have you worked with much or done much research around that?
No, I missed it. Which ones were you mentioning? The ones that are ethanol bound?
Yeah, ethanol-bound cannabinoids. They're not very researched at all. I know CBT in particular has got a lot of medical enthusiasm around it right now, I guess would be the best way to put it. But I was just curious to see if it's something that you had any experience with.
No experience with that. Is that one of the cyclols that is it actual degradation product from one of the cannabinoids, or is it just a rarely expressed one?
I have no idea. I know it's very hard to pull out and stabilize, and that's one of the biggest issues with researching it.
Oh, interesting. Okay. I've been hearing some work on Delta 8. I've been hearing some work from Mark Scaldone as well on some of the hexahydroxy stuff that he's presented, which is all interesting. But a lot of those look like modification events, or just being very careful in how you extract to make sure you can favor one or the other. But I don't have any experience with that particular one.
So you just reminded me of a talk that Mike West did in January. He'll actually be on the show on Thursday. Have you noticed any gene expression differences with plants that were infected with certain pathogens that you knew about? I know he had spoken about how PM plants that have been infected with PM in particular express a new novel cannabinoid that hadn't been previously detected in large quantities before, and I was curious to see if that's anything that you know.
Oh, that's cool. I can completely. So we've done work on not as much on the RNA of what's getting expressed when things get infected, but we've done a lot of sequencing of microbiomes on cannabis. So we have a very large database, and much of this we've published as well. If people want to look it up, it's in F1000. We did some of the first 2 papers on cannabis microbiome sequencing. I think Argonne did one before us on the soil, but we did the flowers. And you can see a very diverse sets of communities of organisms living there. And you can also see this very interesting interplay where certain organisms, when they're present, other harmful ones are not. So we see a really tight correlation whenever there's bacillus or trichoderma, you tend to have a lower CFU count or DNA count of aspergillus and penicillium. So the things that the testing facilities are worried about can be countered with beneficial microbes, but in many states you're not allowed to use them because they also trigger the total yeast and mold and total aerobic counts. So this is something I think we've got to get out to the community, because I'm really nervous that the really broad, non-specific tests we have out there, like total yeast and mold and total aerobic counts, it is really just a testing scam. They're not informative tests. They're just there just so people can rubber stamp, $25 or where they charge, and they're actually screwing you from being able to use beneficial microbes. And it's, go ahead. I think that's creating a genetic bottleneck. I think when you apply that much pressure to cannabis, you're going to screen out all the cultivars that like to work with certain microbes, but they're not allowed to have a high microbial count. Therefore, that cultivar's gone. It likes to have a high microbial count for whatever reason. Maybe it needs it to supplant enough nitrogen or what have you. And when you put a requirement on there being no more than this level of microbes, regardless of whether they're pathogenic or beneficial, you end up screening out all the plants that need those beneficials. And so we've been trying to educate everybody to just get rid of those tests. I mean, we make them only because we have to for certain states, but if the revenue went to zero, we wouldn't care. It'd be the right thing to do. And we could pick it back up on species-specific testing, which we think is the way to go, is target the things that hurt people and measure those. And all the other stuff, let the growers use, because otherwise you're stuck with playing this cat and mouse game with chemicals, people putting on pesticides, then the list has to add 3 more to the list, and then there's another 3. And it's like the Chinese folks making synthetic cannabinoids. The list will never end. And next thing you know, we'll be testing thousands of compounds in the pesticide facilities, and when we can really just use beneficials and knock most of the stuff out.
Mike actually is in chat. He actually chimed in and said it was CBL in particular.
Oh, CBL, yeah, that's the cyclo one, I think. How do you pronounce that? That's the most difficult one to pronounce.
I have not a clue. But that was the one that they found in a lot of
PM infected samples.
Yeah, infected microbial samples. They found that there was a peak in CBL in particular.
So that's interesting, because there is a lot of literature on microbes hydroxylating the tails of the cannabinoids, which I don't, maybe that helps create a precursor for that cyclization event. I don't know. But there is literature on cannabinoids getting modified by microbes. They don't tend to modify the ring structures. They tend to modify the tails and add hydroxyl groups to the tails. So some of the cannabinoid diversity could, in fact, be microbial metabolism of some of the majors.
That's so interesting. So tell us more about how microbial pigeonholing is really leading to serious concerns that you have with the industry, because that's a whole. You brought that up right before we started the episode, and I was blown away, and I would immediately stop you because I wanted you to go into this in length on the podcast for people, everyone else could hear it, because I was just jaw dropped.
So I think the main shocker to us was when we did this work where we did microbial testing the way people did it traditionally. You take cannabis, you wet it in a broth, and then you put it on a plate. But what we did is we looked at the microbiome before it went on the plate, and then after it grew on the plate for like 2 days, and looked at the DNA content. And what we were finding on the total yeast and mold plates was that 60% of the DNA on those plates was bacterial. Now, it could have been endofungal bacteria, which means these bacteria that live inside of fungi, and so they don't necessarily add to the colony count, but they're there. Or they could have been a lawn of bacteria because they grow so damn quickly that the colony is just turned into a lawn, and you see the fungal colonies on top. So we don't know to what extent this is making people miscount the plates. But when you know the plates are that non-specific where they can't discern a fungus from a bacteria, there's no way you can use labs or lacto to try and fight these things off, because they're going to end up training these plates. Now, what happens when a lab tells a grower, you just failed for total yeast and mold when it was really bacteria? Well, they probably go and add fungicides, like myclobutanol. And those things tend to enrich in enrichment in cartridges, right? So here we are, instead of using a benign lactobacillus to take out a mold, you're getting penalized for using these 100-year-old testing techniques of Petri dishes, and then you're signaling back to the growers, the number one fail in cannabis microbials is total yeast and mold. You're signaling them to have mold when they don't, so they end up getting motivated to use fungicides. And chemical fungicides are, I think, much more dangerous to have around because they can enrich an extraction, whereas the microbes are probably going to obliterate them in the extraction process, and they're not going to leave behind. As long as they don't leave behind any mycotoxins, I think we're cool. So many of the beneficial microbes are being discriminated against in the states that mandate using plating. And this is one reason why I prefer the California regs on this, because they're least species specific. They should probably have a couple more species on there because there's things getting through that have visible mold on them. But at the same time, people need to keep in mind that most of the visible mold that you can see in cannabis doesn't culture anyway. Like PM doesn't culture. So when you see a moldy PM, it's going to sail through plating anyway. When you see moldy botrytis, it's going to sail through plating anyway. They're not actually measuring the molds that are allergens very well. And we already know the plating systems are horrendous for picking up aspergillus. It clumps, and they can't quantitatively plate it on these things, and they can't discern the benign ones from the really dangerous ones. And so the molecular techniques are the way to go, and the species-specific techniques are the way to enable these beneficial microbes. The challenge is no one state has it right. You know, we probably need more than 6 microbes in California to do it justice. There's a Cryptococcus outbreak in Bakersfield that gave somebody meningitis. I think we've seen Clostridium botulinum. We don't want that around because that can make Botox and oils show up in a couple states. There's some concern about listeria now from all the kitchen work people are doing, that can end up in some of the milks and be a problem. So there's a couple of these other microbes that are just, if they aren't being addressed, they're probably extraordinarily rare. But I'd rather go after them in a very targeted manner than using these tests that discriminate against vast swaths of genetics of cannabis and aren't really measured in anything related to a clinical risk. Total aerobic count, you know, if you did that with your keys or anything that comes in the supermarket, it'd probably fail all the cannabis tests because they're more stringent on cannabis than they are on things in a grocery store. And it's not giving us any information about if there's an actual clinical risk on the plant.
Uh-oh, I think Steve must just had an interruption.
Yep, I saw him drop out too. So.
Okay, so we can normally go on for probably about 10 minutes, I think, unless he jumps back on here. I think that's how it goes. Oh, he's back. Okay, we're good.
Thank you.
I'm like, whoops. And just in our private chat there, if you want to click on that, you can actually see what chat is saying on YouTube. I forgot to mention that earlier.
Oh, what are they saying about me? Damaging?
Oh, terrible things.
So I had another question. So how does microbial biodiversity, the root system, affect terpene and cannabinoid expression, and as far as the genome goes? Because I've noticed doing aquaponics, when we've done both soil controls, we've done straight DWC, and then we've done split root zones where we had, you know, some soil and some some flood and drain. We've noticed by far the split root zone has a much wider terpene expression and much higher total terpene levels. And I'd love for you to talk about that on how that affects the gene expression.
Yeah, those experiments all need to be done. We don't know it in cannabis yet. I fully believe you're going to find it when you start looking at the nitrogen fixation that's going on in the roots. That's going to be critical for it to build all the building blocks for the terpenes and the cannabinoids. And just this work that's been done in tomatoes, where people are pumping like 6-carbon precursors into the roots, showing it boosts all the terpene production in the plant. So there's already evidence in other plants that don't make huge trichomes that when you stimulate that root zone, you end up getting an altered expression in terpenes. So I think those are great experiments that need to be done. We've done a little bit of sequencing soils. They can be a little bit trickier to survey because there's humic acid in soil, and that can inhibit PCR if you're not careful and doing the right DNA prep. But we've also done some sequencing out of hydros to look for microbial profiles. And of course, they're not sterile. You know, the hydros have all types of microbes in them as well. They're just different ones. And we don't have good correlation on those with terpenes. We do have we've got the microbiome sequence in about 15 strains that also have terpene profiles, but those are all done on flowers. And so we're trying to draw some correlation with the microbe profile in the flowers and whether that is changing the ratio of linalool to maybe terpinolene or something. But we don't have it in the soil area, which is, unfortunately, I'm coming empty-handed to an Aquaponics show here on that.
Oh, that's totally fine. There's always things that we know and don't know. So you were just saying that linalool seems to be associated with terpinolene?
No, I don't. I'm just pulling that out of a hat as to what yeah. But I do think limonene and terpinolene, a couple of them are linked. When we look at the enzymes that make them, it's rare that you get a terpene synthase gene that makes one terpene. It tends to be promiscuous and make a couple based on pH and other conditions. This is stuff John Page has done, where he's cloned a few of these out and demonstrated when you express them in yeast, at least, if you tweak the pH or the input conditions, these enzymes will make a couple different terpenes. And so I think this is why you tend to see in terpene data, there are patterns. There tend to be superclasses, families that you may have seen Mark Lewis present on, where there's clearly like a beta-caryophyllene class, there's a myrcene dominant class, there's a limiting dominant class, there's ocimine, and there's terpenylene, there's a few more. And those dominant classes, I think, are representative of certain genes that are predominantly on in that plant, and they're spitting out a couple terpenes apiece. There is some work that Keith Allen and others are doing at Steep Hill as well that will probably highlight a little bit more of this. They've looked at some gene expression across the Jamaican lion genome, and in particular, just trying to look at the length of the genes and how frequently they're expressed, and whether there are any odd isoforms in the RNA. And so we're just starting to get a picture of that. So I think there's one that we've had a hard time finding. We may have found in Jamaican lion. The gene that makes bisabolol is one that only occurred to us when I think Kyle ran off and blasted that from a gene that makes that in another plant, and he found a sequence very close to that in the Jamaican lion reference. And that is one that's been undocumented to date, as no one really knew what was making that. So just by having the reference, you can start asking these questions like, okay, what's been documented in other plants, and can we find it in these references, and what does that tell us about whether it's being expressed? We have all the RNA expression data for plants that, at least for Jamaican lion's profile, so we know the terpenes that are in that genome that where we don't know where the terpene synthase genes are yet are becoming more obvious because of the RNA work that we've done on it.
So why did you choose Jamaican lion over other cultivars? And is Jamaican lion the only one that's been completely sequenced, or is that the one that you guys have for reference data, or what is the?
Two reasons. So first is in the past, other people have been doing sequencing in cannabis too. So John Page has been doing some great work on Thanola and on Purple Kush, type 3s and type 1s. It references, he sequenced a couple years ago with shorter read lengths, and so the genome is a little bit more fragmented, but it's not his fault. It's just a technology evolution thing. But they usually have about 500 kb N50s in those genomes or less. You want genomes that are more like 5 million base pair N50s. That means the average length of sequence you have is about 5 million letters long, and that's helpful to annotate because genes can sometimes spread beyond 500,000 letters. So if your genome's broken up in 500,000 letter pieces, some of your genes are on 2 different pieces, and that can be hard to manage. But nevertheless, those are really good genomes. So he's been doing type 1s and type 2s or type 3s. Another group, Sunrise Genetics, put out an interesting reference. The Grassa paper, I think theirs was 700 kb N50s, and that was on CBDRX. So that was a genome that was a type 3. So we picked a type 2 because no one had done it yet. And you would get both CBD and THC alleles if you did that. So you'd get arguably more information out of it. But the other reason we picked it was that it looks like a beta-caryophyllene dominant type 2 that makes a THC to CBD ratio challenge the biotech LC patent, mainly because this grower has documentation of it dating back to 2011, if not earlier. Ganja Genetics is the one who made these seeds, and he's done a good job documenting this. And there's won a Cannabis Cup in San Francisco in 2011 with this, and he's maintained the mother line. We've sequenced the mother line, and we also have the seeds that are on sale today at Emerald Farms, and those have popped very similar genetics to what he had many years. So we were doing this to try and get a genome public so anyone who was threatened by that patent could ascertain whether they were within it. We don't know whether this is going to legitimately create prior art that narrows their patent. That's up for attorneys to figure out. But at the very least, we have a reference genome that anyone can look to that predates that patent to say how close they are to something that predates it. So it may become a useful tool if anyone ever gets a letter. They can say, well, more genetically similar to the one that was in 2011 than from yours, and so I think I'm outside of your patent, or maybe I'm inside of it. I don't know. That's all hearsay and speculation on what's going to happen from an IP standpoint. But from a genome standpoint, you want a genome that's both genetics, ideally. And we ideally want genomes from type 1s, type 2s, type 3s, type 4s, and type 5s eventually. So after we put the Jamaican lion genome public in August, that triggered a snowball event where people suddenly wanted to sequence the entire family. And so we got another grant to go and sequence its brother and a bunch of its offspring to do a pan-genome project where we can compare them all and actually look at all of the alleles moving down into the next generation. So when you make a cross with this, we managed to cross the brother with its sister, get offspring, sequence the genomes of those, and now we can begin to see how the alleles actually migrate in their inheritance patterns into the offspring and get a better understanding of how this complex genetic loci is messing with us. Because it doesn't always come out 50-50. You know, you see some of these genomes that are 2-to-1s, and I think that's genetic. Likewise, the 3-to-1s. I think there's a genetic heritability to that, and it has to do with how these genes are in very plastic regions of the genome, such that they don't always have the inheritance pattern we might simply expect from a very simplistic Punnett square that you might see in biology. I think the loci is a little bit more complicated. A simple A-B Punnett square, which is what it's been described as in the past. So all those other genomes are going to help us understand that.
So on that exact note, so I've heard from other, or actually I read an article from a supposed cannabis geneticist saying that polyploids are not possible in cannabis. But I've also seen work from, what was the gentleman who did a bunch of research in the 40s? There was 2 researchers back in the 40s that did a bunch of work with THC and CBD. I can't remember what their names were off the top of my head at the moment. We've talked about them on the show before, but
okay. Okay.
Yeah. So
Adams, I think, is one.
Yeah.
He at least discovered THC. I think back in the 40s before Michelin, and was one of the first to put some of the chemical papers together. But I don't know if he did any breeding on this. But the polyploids have been published. So Jessica Parsons out of Canopy published, or Tweed, I think, published some polyploids, methods for making them, and they've not yet found them to produce more cannabinoids, at least not in that paper. And so they're still, I think, optimizing the technique to see if they produce more yield. I think that they were.
Is that using crocosine?
I don't think they used colchicine. They used another one that's escaping me, but it's similar to colchicine, a very similar metaphase arrest that's less toxic. Used, but the paper just was published about 4 months ago, and so it's out there and has some of the methods on how to do it. So at the moment, they have a method for making these polyploids and quantitating how much of the genome is duplicated, because I don't think you get a perfect tetraploid event. Sometimes you get aneuploidy events, and they're just measuring it by doing staining and flow sorting. So you stain the nuclei, run them through flow sorters, and count sort of the signal from the nuclei as a proxy for how big the genome is. And that's giving them estimates on the picogram sizes of their genomes to make them know that they're bigger than what we see in traditional haploid or diploid plants. I think the other thing that's still unknown is how much these may vary without even using colchicine. We have another program we have going here, sequencing 40 genomes on Illumina, and these are a spectrum of hemp lines and of terpene profile plants, all on short read sequencers. But when we look at that data, we specifically did that work to look for copy number variation in the genome. So it's an unamplified approach. There's no PCR involved, or very few cycles of PCR with tools to count and discount that any artifact that can come from PCR. When we do that, there's preliminarily vast differences between genomes that are in circulation right now. So I think we still don't understand the natural plasticity and variation that exists on a chromosomal scale in cannabis. We could be looking at aneuploids that are out there right now in the rec markets. We just don't have good tools to have ever known that's happening. Polyploids are kind of obvious. Like if you get a tetraploid, the genome doubles. I mean, that's like, you can't miss that. That you can see. But like if the arm of one chromosome doubles, we're not seeing that right now. And we can with some of these sequencers, but there's not been people looking at a whole genome level in cannabis until very recently. So I think that there could be a lot of natural aneuploidy in the population that we're just starting to figure out.
Okay, so one question, when you say that should be obvious, what if it's not obvious?
It may not be obvious with your eyes. You're right.
Okay.
It should be obvious to a cell sorter and some expensive equipment. But yeah, sorry. and it's probably a runt. Most of the polyploids seem to be tetraploids seem to be runts, but other than that, it's yeah, you need some sophisticated gear to figure it out. And sequencers will probably see it, and I think these cell sorters are probably faster and cheaper to give you a sense of like if your genome's doubled. But you know, I don't think these treatments necessarily always result in that. You could get triploids, good hexaploids. I mean, when they do this in other plants, they don't end up with like perfectly replicated, you know, duplicated genomes. They end up with almost like what looks like cancer. And the whole genome's been replicated in many different ways and shuffled, and characterizing that can certainly be a task with sequencing.
Yeah, I always attribute it to being like a dice roll in a role-playing game. You don't know what it's going to give you when you start combining the genome and duplicating. You know, sometimes it's good, sometimes it's bad, sometimes it's both. Sometimes you have really, you know, superpowers, but you also have like no legs. You know, like you never know what you'll get.
I think it's important we get the language of it right, because a lot of people will call that a GMO event. And yeah, the genome's been modified, but you've just made more cannabis genes, right? You haven't put in a foreign gene. Like you're not shoving jellyfish genes in there. It's just messing with the genome, perhaps in ways people have been doing it for many decades.
And for people that don't understand what we're talking about is a polyploid is a cannabis plant where the genomic sequence is tripled or quadrupled using a hormone from what generally speaking, the crocosine, or I mispronounce it, I think you pronounce it colchicine, which is extracted from the bulb of fall crocus flowers, which you can actually buy at the local store to plant in your yard. You know, it's nothing. There's no test tubes and, you know, syringes and you know, high-end equipment. You can do this at your house. Like you can do this in your own kitchen. Like this is nothing, you know, gene splicing or inserting genes, but it is a method of manipulating the genome. So where does genomic editing begin and where does it end when you're talking about something as simple as crocosine versus, you know, that's where it gets real messy when you're trying to regulate this stuff. And I'm glad we got a chance to talk about this tonight, because it is kind of a weird middle ground or gray area.
I think what you're going to start to see are people taking more scarless approaches in GMO because there's such a backlash on GMO. So putting foreign DNA into the plant is one area where people have some concerns over whether it produces allergenic reactions or whether you have off-target effects and therefore you don't know what you're creating. And then what tends to happen with those are more patentable, unfortunately. So there's this motivation people to do that because they'll get patents if they do it, and then therefore they can enforce them on people, and you tend to get monocultures driven this way. It's really bad incentive system. What some people are looking to do is, well, what if you just edit the cannabis genome, like delete some things, don't add in any new DNA, just remove some things. That's in some countries regulated as a different form of GMO. Because you haven't put in any foreign DNA into the plant, you've just mutated it in a little bit more precise manner than randomly waiting for it to occur in nature and pick it out of a cross, right? There's also going to be scarless ways of editing, of moving genes around in the genome, the different parts of the genome, so that they may co-express. And I think you're going to see people do that, and that's, again, going to be one of these questionable editing, like people are popping these genes into yeast, and I'm not going to smoke a yeast joint anytime soon, so I don't think it's going to take over the flower market, but it may appease some pharma people who want pure grade rare cannabinoids that we can't get right now. But there may be ways to move stuff around in the plant itself. I mean, when you look at the whole yeast thing, it's a bit of a parlor trick right now. Very sophisticated, elegant, and beautiful with what they did, but it's one of these scenarios where you're feeding it sugar. 99% of the sugar in the world comes from plant chloroplasts. So, like, what the hell are we doing inviting in this fungi to make this when you're feeding it something that comes out of photosynthesis? Once cannabis gets outdoors and we're not having to grow it inside of prisons, the price is going to collapse. And we're probably going to have a better job breeding for better photosynthesis to make sugar, to make cannabinoids more effectively right there in the organism where they came from. cannabis is a carbon sink. Yeast puts out CO2, right? So we've got the whole carbon thing is backwards too with it. So I'm not giving up on the. I think the plant ultimately may win that battle, and where you're going to see the yeast take off is making these purified compounds or these really weird modified ones the plant can't even do. One thing that Jay Kiesling showed in that paper is they could feed it some weird hydrocarbons and it would make cannabinoids we've never seen before.
Yeah, but the problem is, and there was a medical study in was it Spain or Portugal that they did, and they had 32 patients and 29 of them died when they used a novel cannabinoid that they were.
Oh, yeah, absolutely.
You know,
it's dangerous.
So and that's what worries me too with the genomic editing and adding terpenes, and to a lesser extent when they fold some of these floral terpenes in with cannabinoid terpenes, you don't know what that combination is going to do. Like, that is literally, in my opinion, dangerous because we have such little understanding of that.
I'm glad you said that because I get ridiculed for some of the same thing. The thing that worried me most about Kiesling's work is they put it in cerevisiae. This is a brewer's yeast, and I guarantee you there have been stabs taken of that into every college dorm that had their fingers near those strains. If you have a strain at a university and it's known to make cannabinoids, I do not think that's been retained inside their freezer perfectly. I wouldn't be surprised if people have now. So now you've got a strain that can make cannabinoids, great, but what if you feed it these alternate precursors that make some crazy cannabinoid like HU210 that has 100 times more potency? And the students don't know this, and they muck with it, and you could have some unexpected, acid tests, if you will.
Is that THCO, the weaponized one they worked with in the 50s? The THC acetate?
No. HU210 comes out of Hebrew University and is one that is much more potent on the CB1 receptor. I think Mitchullam has some background on that. It has HU in front of it, usually from this lab. The ones that have turned into a lot of the synthetic cannabinoids, unfortunately have JW in front of them, which stands for the gentleman Huffman, I think, JHW, who made those compounds. And then Makrianis has all the AM compounds. Alexandros Makrianis out of Northeastern.
Now, have you?
One of the Tylenol things is there.
Have you worked at all with things that increase that change the way your CB1 and CB2 receptors react? I know I've done a lot of work with a couple of different compounds.
I have been involved in a. We did one study that we published that did 6,000 patients that we sequenced that had epilepsy and mito and autism, and just scanned the genomes to look for mutations in the cannabinoid receptors, and CNR1, CNR2, DAGLA, FA, MAGL, and we did find some hits. There were some mutations in the cannabinoid 1 receptor that were tightly associated with migraines and anxiety, and there were mutations in DAGLA that were tightly correlated with seizures. And so these have been published in the rare variants that if you have these mutations, you probably have a genetic predisposition to endocannabinoid deficiency syndrome or something, but not anything from a chemical standpoint. We haven't done anything trying to modulate the receptors. It's not in our bailiwick.
No, that's totally fine. We had a question from Chad is, what is the safety, and we were just kind of talking about this, but what is the safety of CRISPR or Cas9 or lack thereof regarding, now that you can buy a CRISPR set for less than $5,000, what is the potential chance of someone getting real crazy with their, in their garage with cannabis and cannabis genetics?
I think it's already happening, is my guess, just because those tools are so easy now. And I've seen a couple things on Instagram and other places where people have performed agrobacterium transformation. It's been published before as well. There's been published how to do agrobacterium transformation. So it was very hard to do without good references. Now we can be more targeted about it. The genomes are in near chromosomal shape, so there is some off-targeting risks that people need to be a bit concerned about. It's better, if they're doing it, it's better to go and verify where the hell you made the edit, catalog that so that people can at least track it. That would be a very good use for blockchain, because I think if people do make those edits, it would be nice that if they publicly disclose what they are. So if we ever see them floating around in the pollen pools and fertilizing other farmers' lawns, there aren't lawsuits that emerge due to false pollination of IP protected, CRISPR stuff. That does happen with the Monsanto genetics. they get blown around and people get sued because they inadvertently got pollinated by someone else's patented crap. So I've heard of folks knocking out THC synthase with CRISPR, so they have 400 to 1 CBD lines. I've not seen it published in peer review. I've just heard rumors of it, and I think it's coming. And unfortunately, I wish it weren't coming with IP around it, but I bet that's going to be the main attraction for people to do this, is that it'll be hence man-modified, much more protectable, and it will give them more funding, and therefore there will be people making probably any edit they can to secure IP. Whether it is beneficial or not in the long run is probably going to take some time for us.
So on that exact note, what is the best way for breeders currently, given the current fiascos and other things that have gone on, what is the best way for a breeder to use a service either like yourself or someone else, or even their own genetic testing abilities through whatever their resources are, to protect their IP and protect themselves and protect their own, a lot of these people have been breeding for 10, 20, 30, 40, 50 years, or even have lineages that have, their parents and grandparents were growing them. I know I can speak for a lot of California, at least, where I've met a lot of these people that have 2, 3, 4 generations going back.
Yeah.
What is the best way for them to get their DNA sequence for their cultivars and to protect them in a way that best helps them? Because everyone's afraid right now. Everyone's terrified. Everyone feels really, backstabbed by some of the things that happen.
Yeah.
What is the way forward? What is the way that they can use the type of data that is available to them to best protect themselves and to best, further help the industry as well?
So Dale Hunt has some good writing on this, and it really comes down to, I think, budgets and costs. I think in the past we have been perhaps not good advisors in that. Okay, offensive patents are really expensive, so they're 20 grand. No one's going to do that. So the only thing you can do is defensive, and then you can do sequencing to maybe help that. That's probably not the best advice anymore, because some things have changed. Sequencing prices are cheaper, for one thing, but two, there's also ways. So there's probably a tier. If you're like below a couple thousand, I think he wrote this up. If you're below like a couple $1,000s, and that's your budget range for this, probably defensive IP is your best play. Is get your documentation, and whether that includes DNA sequence or not is not necessary. You want to have documentation from the timestamps of when you have this material growing, and any evidence that is well documented from a notary standpoint, if possible, or something where it's an undisputable time reference, like you were in a cannabis cup and you won an award with a particular strain, and you can tie that to the strain that you had. Those are important to collect, so you have an evidence train of when this strain existed, so you don't get submarined by patents that are about to emerge. That, I think, is step one. If you don't have that documentation, well, timestamp, go and get it. Terpene profiles are a good start. Sequencing isn't necessary to file, but it's a great, I think it's going to be very helpful in the future, and that's going to be the language the USPTO uses to figure out whether something's a new varietal. Then when you get into the $2,000 range, that's the price it takes to file a provisional. You could file provisionals or go the PVP route. But now you're starting to get in the range where you might want to get someone like Dale or a cannabis lawyer involved to figure out what's the right thing to do with your role in your circumstance. But you can do provisionals even without DNA sequence for a couple grand. Challenge with a provisional is in about a year, you're going to be forced to decide whether you want to go 20 grand deeper into it. And some people at that point decide to say, screw it, I can't afford that. I'm just going to put it public and go defensive on this. And forget the full filing. Other people go the full filing. So I think he's tried to break it out into 3 different cost buckets as to, the real, cheap ass way to do it, where it's more open source, the file original and see what happens path, and then also the full boat, like trying to do something, that may in fact be a full patent, utility patent. There's also a couple other angles here outside of utility patents, which are a bit more expensive. There's Plant Variety Protection Act, which everyone should into, and then there's also the USDA is now opening their doors to do hemp that way, where you can send in thousands of hemp seeds. They pop a few to see how diverse they are and that they're in fact uniform, and then they might give you a plant for Protection Act. And then there's a couple other ideas people are kicking around that are more open source based ways of trying to defend against this. Now, when it comes to sequencing, the only thing I would say, having been in the sequencing space for a long time, is don't trust anybody. You want a scenario where you have the keys to your data, and that the most important thing you get isn't some fancy star in a galaxy, but your actual sequence data is what you need to convince the USPTO that you have a fingerprint. They do not understand the coordinate system of the galaxy. I don't understand the coordinate system of the galaxy. So when I see dots up there, I can't tell you whether another strain I have in my hand matches any one of those dots without the DNA sequence information. And I think that's what people were so frustrated with them about, is they held that private for 2 years while putting stars in a galaxy that weren't real prior art. But that's the information that if you do go down the road, make sure that when anyone does your sequencing, they deliver your sequence files, your FASTQ files to you, and that you have access to those. You get to decide whether they go public or private. Ask them about their data release policies and whether or not they're going to utilize your data for other harvesting. That's possible to happen. Even in our case, we may choose to go look through the data to find other markers. We've not yet found markers in the stuff people have sent in. When we've had to hunt for markers, we've had to have very specific programs that look at, in the case of the CBD gene, we had to go and hunt down CBD genetics and THC genetics and run our own study because we couldn't wait for them to randomly come in the door and hopefully answer the questions, because not everyone was giving us the chemotypes. So we just did our own private study and figured that out. We did the same thing with XY testing, as we went and hunted down males and sequenced their genomes to figure that out. We didn't try to randomly figure that out with people sending stuff in. It's not very efficient to do that. But nevertheless, you want to know what they have access and the ability to do and look at their data policies. But we really do think the blockchains are critical, because those are the only timestamps we can't edit. If we disappear, your data has been hashed on those blockchains, and that timestamp is immutable, is publicly distributed. And if we disappear, you can still prove the time at which your sequence existed without us being around anymore. And I think those really help.
It's kind of like an poor man's patent.
Yeah, like putting it in the mail with a timestamp, right?
Right.
You just can't do that for the USPTO. So we print it on something the U.S. government can't shut down.
Email it to yourself.
Yeah. Though emails, I think might get challenged now because people can edit some of that stuff. But who knows? We'll see. All we know right now in terms of blockchains and IP is the Supreme Court of China just accepted blockchains as valid notarization. It's not necessarily given an official okay here in the States, but we are seeing a lot of the patent attorneys are talking about integrating blockchains into the discovery mechanism and documentation process. So I think you'll see will get adopted. They're being used in banks, and they're being used to prove the timestamps of who owns what on billions of dollars of money right now. So they're trustworthy.
So there's a lot of talk with all the different patent stuff. How far do you think this is going to go, in practicality, considering the fact that there's a huge pool of genetics that have, pre-existed a lot of these patents that are trying to be claimed, especially some of the ridiculous utility patents, which I'm happy to say ridiculous because I've seen some of these, I've read some of them, and some of them are completely ludicrous. And you mentioned a couple earlier. Where is this going to actually realistically shake out? It's one thing to actually file patents and do all this other crazy stuff. How much is this actually going to matter in, say, 1 year or 2 years or 5 years down the line? Everyone already has their genetics. They've been growing for years and years. If I'm a commercial producer and I've been growing something for 5, 6, 7 years, how's that going to hold up in court realistically when they come and challenge me that I'm selling something that they have a claim to, when I can go back, before they even, that it gets so kind of wonky and ludicrous to me that how is this going to realistically shake out? Because you've actually worked in a field with genomics and a lot of other things, in real practicality, where there was this kind of scenario, maybe not quite like cannabis. No, there's never been a scenario like cannabis, but in a similar kind of vein with the genetic kind of stuff. And where is this going to realistically kind of fall out in a kind of a real world scenario versus what everyone's trying to do right now?
So there's a couple answers to that. Some are good news and some are bad. The good news is I don't think people like Biotech LLC are going to be going after people who are doing craft grows. I think when they have that patent, it's issued now in the States, but they're not going to go after anyone in the States, I think, right now because of the federal overhang. There's a risk that the federal government could just kill the patent because of the illegality of it. So they'll probably go to Canada first and find some LP up there that happens to be growing a strain that already has a shitload of inventory of that strain that matches their claim and say, hey, why don't you give us a piece of that? Because it's going to cost you more money to invalidate our patent than we're going to ask you for a royalty. The invalidation process, there is a presumption of validity once a patent is issued. And that's something that is really important in the discussion because to reverse an issued patent, you need an ex parte re-exam, which can cost hundreds of dollars to execute and take years. So while you may have strains that you were growing that match those claims from 5 years ago, unless you've got really good documentation proving those timestamps that you were growing that many years ago, the presumption of validity is not going to be in your favor. And that means you're going to have to go through the re-invalidation process of the patent, and that's going to be enough money to make you want to just pay them off. The good I don't think they're going to come after U.S. growers, and they're not going to go after small ones. They're going to find some big publicly traded LP that has money they can throw around and doesn't want to see an enormous lot of their strain disappear, and they're going to look at $100,000 to $300,000 multi-year endeavor to invalidate the patent, or pay these guys 50 grand to get them the hell out of the door. I think you're going to see more of the latter, and that may in fact feed a frenzy for other people to try and do the same thing by getting more genetics issued, but. I think a good thing to do would be to keep an eye on the USPTO. You can search the USPTO for cannabis patents, monthly, and just see which new ones are issuing and how many of them are plant-based and how many of them are for some of these crazy. I see there's one right now being contested in Colorado over a 95% CBD formulation or something, which seems very obvious to many people in the field, and get it issued, and people are now having to fight it. So, I think it's a little. I get a little nervous when I hear people say, "Oh, everything's already in the public domain." It's like. It's not unless you documented it. And it's not unless you documented it in a way that the USPTO is used to measuring these things. And they're not used to looking at pictures necessarily on Leafly as being informative for differentiating a novel strain from another. They're used to looking at, I think the best thing you can do is look at what they've issued. And that's the best proxy. And what they're issuing are things that talk about chemotypes and genotypes. And so that's the language they want to see to differentiate these things. And that's probably the language you're going to see the future patents that come out. I think the equatorial sativa one, which is a plant patent, is talking about having 10-fold more limonene than another varietal. And so it's been differentiated, but just on having a really, really high terpene profile.
My question is how realistically could you defend a utility patent if you don't include the cultivation method to the brutal level
well,
of
that's
to get that application of terpene expression?
So Mark in his patent, to his credit, does a very good job looking at the variation in terpene expression he gets under his conditions. I think the big unknown is that they deposited seeds to a foreign seed bank that no one can get access to without breaking U.S. law, getting them back in here. So in order for you, what the really million-dollar question of the patent is, if you pop 10 of those seeds, do you get 10 different varietals? And if you do, then it's very difficult for them to apply the argument that, well, look, the Jamaican lion you sequenced wasn't a back-crossed homozygous clonally replicated seed. It was an offspring, and so it's not pertinent to what was around in 2011. Well, if they're using a seed to get into a bank, and we're using seed from a seed derivative of something from 2011, that argument may not go both ways, right? So they hopefully the seeds if the seeds they submitted aren't stabilized, I think they're going to have a really hard time enforcing the patent.
So on a genetic level, what is the F level to actually achieve stability in cannabis? You're the person to ask as far as genetic goes. What is because this is a question I've often asked people that want to tell. This is my challenge question to people that when they want to go and patent a strain is, okay, what is the standardized F level? How many times do I need to back process or stabilize it before I can actually claim that this is a stabilized strain? Because has that even been standardized in cannabis? Is there even an answer to that question? And if so, what is the answer?
I think everything's theoretical. I've been seeing theoretical calculations that are between 6 and 12 back crosses, but I think it needs to be done. And I think a lot of that depends on how distant you're starting. So if your parents are as far apart on the genetic spectrum as possible, when you cross them, it's like mixing chimpanzees and humans. You're going to get a really wide spectrum, and to get those back to homozygosity, you're going to have to cross them probably more than 10 times to do that. If your parents are fairly closely related, you can probably achieve it in faster back crossing. But again, this is all. I think these experiments we need to be knowing and doing right now is actually run those types of sequencing experiments on multiple back crosses. See, all right, how much of the genome actually stabilizes.
Because that was the very first guy that I can remember trying to patent a strain was Big Buddha Seeds wanting to patent the cheese strain, even though we all know he didn't invent cheese. And my question was really, because what F level have you bred it out to? And you know, standardized in most agriculture, it's what, an F12 or an F8.
Yeah. Yep.
For most agricultural crops, and it's so wouldn't cannabis just be another it's a plant? Why would that be different than normal agricultural crop? It should be out to those same levels. So you can't just go and claim an F1, an F2, or an F3 is a new cultivar that you invented. You actually have to stabilize it to the point, which you've talked about a little bit, but trying to break it down into, everyone's familiar with F levels, especially less educated breeders or people that haven't quite gotten into the genomic end of it. And that's one of my question to people that want to talk about patents is, okay, what is your agreed upon F level that you think is actually when it's stabilized? And this is again the question that if we're going to actually start doing patents and throwing money around or trying to take away money from other people, this has to be put into federal law, because until that happens, there really isn't a standard. And it's going to keep going back and forth, and you can have a real slick lawyer totally overturn a whole bunch of stuff.
Yeah, and there have been so it's a good question. I don't think we know the answer yet in cannabis as to what F level you need to go to. It is, but you're right in that there's reason to believe it should be as far, if not further, than other plants, just due to its rate. It's a very polymorphic plant, which means you need to do more back crossing to actually achieve a stabilized line. So that's an important point. There has been some benefit in Canada. There was a group of people that threw a bunch of money together in opposition to the patent, and so they managed to shrink some of the claims in Canada for at least the biotech LLC patent. So there are some motions being made out there in the field to push back a little bit on that. That's at least reassuring to see.
All right, well, we've kept you most of the evening. I don't want to keep you the whole night. I am extremely grateful for all the immense wealth of knowledge you've dropped on us, and you're more than welcome to come back anytime. I really appreciate you taking the time to share your knowledge with us, and bring back a level of authenticity and fact checking to a lot of the different controversies that have gone on within our community. And I very much appreciate you taking the time to do so.
Sure, thank you very much as well. And if anyone wants to connect with me on any of these other offline questions, I'm on Instagram, and we've got a Facebook page and LinkedIn, so you can find me there. If not, medicinalgenomics.com is where we house Cannapedia.
Well, wonderful. I have a link to your website as well in the description. That'll be up both the audio and video versions of the broadcast as well.
Great. All right. Well, thank you very much.
Thank you so much.
Enjoy it, Kevin.
All right. Take care.
Thank you.
Cheers. Yeah. Is that all wrapped up?
No, you're welcome to hang out if you want to.
We'll talk about our grows and stuff, but you're more than welcome to take off.
Yeah, all right. I might run in 5 minutes or so. I totally get off 11:30. But I love the lava lamp you got there in the back. I need to spice up my background.
Oh well, hey, it's a YouTube thing.
yeah, it works.
Tara's hippie den.
Yeah, my little canvas cave.
So are you guys doing any work to measure the microbiomes inside the aquaponics area? That'd be really interesting.
Yeah. So we actually I was working with a vegetable farm, and they actually were part of a NASA study where they tested, I believe it was 52 different aquaponics farms, and then they were trying to figure out what microbes are responsible for mineralization of different nutrients across a wide variety of different things. Because if they're going to go to Mars, what microbes they need to bring with them. And they tested all the soil.
Oh, man.
And they did a bunch of organic soil growers. And what they found was, on average, there was 168% more biodiversity in terms of different species in the aquatic environments and the terrestrial environments. And they were much more complex nutrient chains microbially. And that specific study was used for evidence against when the organic soil farmers were trying to say that organic hydro and aquaponics should be disallowed. They actually used that NASA study as evidence as to why they should be allowed, because they actually had more, complex mineralization chains than the soil did even.
Was this with the JPL by chance? The Jet Propulsion Laboratory? Did they fund this, or is it...
I don't remember. There were some people that came and took some samples, and then they sent us an email a couple months later. So
I had this in a former life at Agincourt, when we were one of these sequencing facilities. We got a contract from the JPL. And it's an interesting story that taught me a lot about microbes, which what they were doing was swabbing every part of the microbe of the Mars rover to catalog what life was on it, and then irradiate it so they could try to send they didn't want to contaminate Mars and destroy the life on Mars question by dropping a rover up there. So they had us sequencing this stuff, and then they were using a variety of different techniques to try and sterilize it. And they would repeat this over and over again. They'd send us samples, and we keep sequencing. We didn't know what they were doing. We just knew that they were trying to get the thing clean enough to send up there. And what we noticed throughout the course of the sequencing is the diversity was collapsing, but the bugs that were getting left behind were sporulating. There are things that like anthracis, the very bugs that would probably survive the trip to Mars, is what they started enriching for. And so they backed off at the end of the project and realized, we're never going to get this thing to be 100% sterile on launch. We just need to catalog the hell out of it. And the best thing probably for the flight to Mars are the very bugs we tried to remove. Like, if they just left the earthborne microbes on there and didn't irradiate the hell out of them, they wouldn't have enriched for microbes that could survive radiation. So they ended up backing out of that, being like, we're just going to let it be all contaminated, and then we'll let the other microbes that, like, usually on your hands are probably eating all the ones that would sporulate, so that when it does fly, it'll get irradiated appropriately, and we won't have this living selection thing going on. So that was a really fun project, because they ended up pivoting halfway through it, being like, forget it, we can't possibly make it perfectly clean enough. We're just going to catalog what the hell we put up there. So if anyone ever does claim life on Mars, they'll have to compare it against the earthborne database that may have contaminated the rover and say yes or no. But it's kind of a lesson, I think, for the cannabis field, because there's a lot of irradiation going on right now. They're radiating flowers. They're radiating for lowering the microbial content, and I'm worried that those radiation programs aren't being run in a way where they can pick up what we saw on this rover. Mainly because they're mostly measuring whether their radiation was successful by using Petri films. And the Petri films, they'll get the epiphytes, the stuff that grows on the surface of the plant. You can get to go on a film. The endophytes that live in the plant, like, which are the ones that do the magic in my mind, is like aspergillus and botrytis and fusarium. All these things are internal. If you want to get those things, if you want to be able to quantitate those, you can't do it with plating, because you have to crack open plant cells to get to them, and the act of cracking open the plant cells cracks them open. Right, so the concept of using plating to figure out and measure endophytes just doesn't work. And we know this in the microbial world, that if you want to get to the stuff in the plant, you got to crack open cells, which means nothing grows. And so you can't count them on Petri films. You got to count them with PCR. Anyway, we're involved in some studies right now with people who are doing irradiation programs, and we're trying to show this to them, that your irradiation isn't actually killing the endophytes. You just can't measure them on plates. You think you're you're getting it sterile, but you're not. And you might not want to do it this way. You might want to just use bacillus or some other microbes that get rid of these things, because it's going to be much harder to kill endophytes. They just have multiple nuclei. They're much more infectious. They're harder to get rid of. It's better to get rid of them with other biological weapons than gamma rays.
I'm kind of blown away that you said the government they actually had some forethought to think about not contaminating Mars. That's
I'm stunned too.
Right there, I went, what? They actually thought ahead for a change instead of
the guys.
You know,
I got a card from the guy. His title was the Director of Planetary Protection. Like I asked him for a second card. I'm like, that's the coolest name in the world. I wanted another one of the.
It's like a new sci-fi movie or something, you know.
Yeah, but they still do great work. They're still publishing all types of microbial work from the space station now. Like the microbes that are growing on the space station are some of the same microbes we're finding in cannabis. They're having Ralstonia infections. All the things that plague some of these indoor grows are up in the space station. So there's a lot to learn from whatever they're doing up there and what we're doing in these simulated growth environments down in Canada.
We've come a long way from NASA using GH3 part. That's all I can say, you know. Yeah.
What's that?
We've come a long way from NASA using general hydroponics 3 part for growing, you know, their hydroponics and all of that.
Oh, yeah, that's right.
And now they're into aquaponics, and they published a paper. I think that's awesome because when I was first starting, I was always one of the reasons I studied and used 3 part for a long time was because NASA used it. You know, that's what they started.
Nice, yeah.
And so now that when you say, well, one of the leading papers, you know, that got, you know, where they were saying no, aquaponics and organic hydroponics should be used because of all the extra microbes and all it provides. I thought that's a long way. I mean, that's a long way in about 15, 20, 30 years, you know, that they've come. Anyway.
We had a question. Have you tested any genomes from plants that have been in space, and how would that affect cannabis?
We've got to get Elon Musk to do that. Right? SpaceX mission number 1, grow a pot plant up in space. No, we've not. All we do know they've run this stuff in space, but not on cannabis DNA. They studied some microbial contamination with one of these devices and these colorimetric tests up in space. But now, I'm kind of curious what would happen in a micro or zero gravity environment. Like, which direction would the root tap go?
Cannabinoids, man, space cannabinoids.
Yeah, there's just too much fun you could have with that project. I think it's all going to burn up on re-entry. I mean, you're going to probably burn too hot on the way in, and you're going to get CBN. Bad re-entry for the plant. All right, with that, I think I gotta check out. I just realized it's almost midnight here.
Well, I appreciate you taking the time to educate all of us, and you're more than welcome to come on any other time. If you got something you want to share or educate people on, please let me know. You have an open platform to do so.
Oh, thank you so much. Appreciate it, everybody. Take care, guys.
Thank you, Kevin. Good night.
See you, Tara. All right, bye.
Cheers. Thank you. All right, that was an amazing episode. You can check out more of his work over at medicinalgenomics.com. They do sex testing, they do genomic mapping, everything that Phylos did, but in a much more ethical way. So definitely check him out. And as you can tell, he's as opinionated on Phylos as I am. So that was pretty fun. That was an amazing interview. So much love, and thanks for him to come on. What's up with you and your grow, Roger?
Oh, same old. We're going in the middle of flower. Everything's going real good. Oh man, I went in last night and finally got buds where stick my nose in it and you can't get the smell out for like 20 or 30 minutes. So I got some sweet stuff going on in there, but not much else. So it's been 100 degrees 3 days. We talked about weather, and I was worried about you because it looks like I'm surprised the state of Oklahoma is still there with all the newscasts about flooding and how bad it is out there. But, you know, I felt like, you know, it's funny, we're on this weather tip. We've had 3 100-degree days in a row here out of the blue. I mean, we went from fall to spring with no winter, basically. And now it's been 100 degrees 3 days in a row. I walked across the yard a while ago on the way here because well, it was so hot today that at the end of the day, I went and took a nap and I slept past the start of the show. And I woke up and I looked at the clock and I said, oh crap. So I let the dogs out and I come across and it was kind of like I stepped out and it felt kind of cool. And I said, well, that's weird. 100 degrees today and it's so cool. And then I started walking. I walked across the driveway to my office side, the farm side, and I stepped down off the driveway, and it felt like I stepped into a heater. You know, just a weird effect where I went from about like 70 degrees to like 80 something in 2 steps. And so yeah, it's pretty hot out there right now. My plants, I didn't realize, I didn't really keep up with the, you know, because it's been so weird and fairly mild even for us, you know, because the average, well, the newscasters or the weatherman actually said yesterday the average temperature here normal is 85 at this time of year. And we've had 3 days at 100. And I didn't even notice it a couple of days ago. I went out there and they didn't die or anything, but my pepper plants and tomato plants looked rough. So I went and dug out all my shade cloth and laid it out so I can put it up. I might have to put it up tomorrow because they're not going to be able to survive if it keeps this kind of, you know, heat up. All my produce will be, you know, fried out, you know. So other than that, not a whole lot. It's like, that's basically all I wanted to say. It's been really super hot here, and we were worried about you know, out there in Oklahoma, because we're getting all kinds of news here on the East Coast about, you know, but like I said, I'm surprised there's still a state there that hadn't washed away yet. So
yeah, glad you're doing well. Exactly. Sorry, I had to go put my chickens away. I almost forgot.
Chicken.
My regenerative. Hey, get my regenerative going. My closed loop.
Yeah, I want to get chickens myself, Tara. I mean, I've been wanting to do it for a while. My brother's got them down the road, but we're not supposed to have them in our neighborhood, even though it's a country neighborhood. But my neighbors got them, and they put their damn chicken coop. They built a new chicken coop now. All of a sudden, used to be behind their house, which was like 250 feet away. Now they're right across the property line. So I hear them freaking chickens all damn night. You know?
I will tell you, if you do live in town and you do have neighbors, do not get a rooster. They crow at all hours. They will, like, they really.
Yeah. Right.
I have 4 of them, and I'm like, I got tired of them. I'm like sick of them crowing all night.
Yeah, there's none of this. They only crow when it's sunlight, when it's time to get up. Them some bitches crow all freaking night. Well, it kind of starts after midnight. It is after. It seems like it is after midnight.
My husband says it's at 3 o'clock, and it wakes him up every night. He says, because I like to sleep with the window open. I like fresh air, you know.
Yeah.
And yeah, he says every night. I'm like, well, sorry, you know. Yes, it was an awesome episode. I agree. Thanks everybody for being in chat and hanging out and all the great questions. There was amazing questions tonight too. So
well, it was busy. I went in and looked at that earlier, and it was like I said, because of my eyes, and every time you try to scroll up, somebody's commenting at the bottom, and it's just moving. It was real busy in chat tonight for sure.
It was, everybody,
you guys.
Go back and rewatch it. There was like, woo, so much information. Like, this will definitely be one of those I'll put on a list and I will rewatch, and like, yeah, really good episode.
Yeah, it's like rewatch the episode on the left hand and Google on the right hand.
The first episodes where I go back and re-listen to. It's not where I'm actually listening, because you know, I go back and sometimes I'll do some minor edits when we reload the audio version, but this was a really info deep episode. It was really good.
Oh yeah, very interesting. Very much. You know, I'm sorry I missed the beginning. I'm gonna have to go back and listen to it just to get the first episode.
So little update on my situation. The flood waters are about 220 feet from the greenhouse right now, that we've been working so hard on. The water is supposed to rise another 1.5 feet about by Thursday, assuming we don't get more rain, which we got 2 rounds of heavy rain today. So we'll see what happens. We're supposed to rain all day tomorrow, so that'll be interesting. So we'll see if it actually makes it up to the greenhouse or not. But we're on a high point. We're not even like we're not. It's crazy to see how literally it's probably 2 miles wide where we're at, and this is a stream that normally I could swim across and probably, I don't know, maybe a dozen strokes doing butterfly, just overhand, left, right, going across.
Yeah, swimming. Yeah,
swimming. Yeah. Like a normal, again, like maybe 10 or 12 strokes, and you'd be across the river.
Normal thing for there? Like, I don't know that way.
No. Normally, there's a 30 to 45 foot, depending on the area, cliff that you could jump off of to go swimming in this deep hole. And it's not only is that filled, it's another 20 feet above that. It's insane.
Freaking nature thing, like they don't have flooding there normally and stuff.
No. This is the worst flooding they've ever had since they put most of the flood control in the state of Oklahoma.
Oh my gosh, I didn't know that, Steve. My gosh, geez.
Are y'all doing anything with sandbags around the greenhouse? Are you trying to do that?
Oh, thankfully, so we just got a transformer installed and everything for the electrical, but it's not tied into the poles yet. So we're not going to tie it into the poles.
Good for you.
Because I don't, and we sandbag the electrical box so that, if there is a flood, that's going to stay dry. And everything else, there's nothing else really to worry about. All the outlets we put on the above, they're all up on the rafters. So, it's all the other electrical outlets that could actually, be an issue are all facing downward, about 10 feet above the ground, so that's not going to flood. If that goes underwater, we have much bigger issues.
If you have a forklift though, and you have access to those big feeder bags or whatever, those big totes, you can get those totes sometimes for like $7 to $5.
Yep.
And you can fill those with sand, and those work really well. I see him using them all up in the floodwaters up above us.
Yeah, well, we really, again, because we're kind of mid-construction, it's not really a big deal for the water to flow through the frame of the greenhouse. We don't have the plumbing in the ground yet. We're just finishing up getting the light deprivation put in this week. Next week, we're going to start cutting assuming the light dep gets finished this week, depending on flooding. They'll start cutting in the plumbing next week while I'm away teaching with Josh. But that's where we're at right now. We're just with as much rain as we're getting. It's supposed to rain today and tomorrow, and we're supposed to get rain every day next week. So it's kind of insane.
Wow, jeez, we've actually had a lot of rain too here, but not like that.
Yeah, that's far. We got a week 100-degree weather with no rain at all over here. We're in Swamp City, so that's amazing how. Yeah, but there's no climate change going on, not at all.
Yeah, we just hit 84 today. It went from having to have the heater on over here to now I have the AC on over here. And then I got cold. So, yeah.
Sounds like dragonflies. They've had the same problem.
Yeah. It's all over the area. It's crazy. It's just went from hot to cold. We're used to having a little bit more moderate, gradual into that summer. But oh my God, I went outside and took me away. So yeah, I think I'll be planting in my beds.
Yeah, we've just been ordering soil for our outdoor grow. Whenever this water subsides, we'll get that going, and then just prepping everything. We have a whole bunch of plants ready to go in the greenhouse. We're about 3 weeks behind because of the insane rains and just delays. Like currently, the road to our greenhouse is officially closed.
Right.
But you can get to the greenhouse before you hit the river, so that's fine. We have
So where y'all did the show was the bedroom, right? Isn't that what it is? It was the bedroom where you did the show, where the guys did the show originally?
We have 2 separate cultivation properties.
Right.
And the property that I showed you where I was filming before, the road to get there normally has been completely destroyed. The water has been too high to repair it. So they had to take down the fence in the back of their property and basically create a new road through their neighbor's property, who thankfully they're really cool neighbors. And that connects out to a wholly different road that you normally have access to the house. And that's how we're currently getting to that greenhouse. But we do have a road access to get there from where we're at, so it's no big deal. But it took us literally a day probably to take fencing down and to bring in a couple of truckloads of gravel to get the road built to where it was, because the property is completely flooded out on 3 sides. It's on a high point, but it's surrounded by water on 3 sides. So it's, the neighborhood is, so that's what we got to do right now. We're just trying to keep everything going, make sure we don't lose any plants. We have an insane amount of plants that are ready to go. I'd have to take count of how many cultivars, probably 80 different cultivars. We're going to grow a bunch inside and a bunch outside. We're going to do a bunch of side-by-sides, over 50 side-by-sides with soil versus aquaponics, and get an immense amount of data on what expresses better in soil versus aquaponics. And it won't be through one cultivar. It'll be through a wide variety of cultivars. And they'll all be clones off the same parentage. So we'll get really, really good data off of that, which I'm super stoked on, which is going to be one of the largest projects to date. Certainly the largest project in a side-by-side for aquaponics versus soil. We won't be able to compare yield data because the planting dates won't be the same, but we'll absolutely be able to compare chemovar data, which is, in my opinion, vastly more important anyway. That we'll have different grow times.
Yeah.
The veg time will be different.
So cool to see, because awesome, firsthand right here. Cool.
One of the most exciting things about me moving out here was the ability to test, hundreds and hundreds of plants in the side-by-side. We're going to have over 800 plants outside, and we'll have about 3,000 inside. We're still working on where the fence line is going to go. We're actually using this flooding right now as we're never going to plant anywhere that's flooded right now. So we were going to put a hemp field down in the lower area. We're going to completely move that to a different area of the property. We have 1,800 acres to work with, so we can just move stuff around. It was almost a blessing in a certain way that we didn't go ahead and plant out the field of hemp that we originally intended to. We ended up with some delays, mostly due to weather, and it's a blessing that we did, because otherwise we would have lost thousands and thousands of dollars. We would have lost to flooding. We would have lost a whole field worth of hemp, acres and acres, if we had planted out where we had originally intended. So, it was a good thing that we didn't, but a great thing for next season. You know what I mean?
Yeah, exactly. So, hey, where is Dutch at? Have you talked to Dutch Blooms and Regenerative, or where are you guys at on that?
Oh yeah, actually, I want to mention one other thing with the flooding. So, I actually was talking to a gentleman about this over the weekend. If you are dealing with flooding, be it Kansas, Oklahoma, Mississippi, Louisiana, Missouri, anywhere on the Mississippi Delta right now, or the river basin is completely out of its banks. If you want a way to clean up that property and actually grow things without dealing with pathogens, because remember, when you have floodwaters, you have an increased chance of E. coli, salmonella, listeria, all kinds of things that could be a liability. If I'm trying to grow a farmer and I want to grow a quick crop, say squash or melons or something, they basically can't grow that this year. You can get around that if you apply lactobacillus ferment, and it's not very, extremely cheap, but lactobacillus loves to feed on E. coli, salmonella, listeria, other human-borne pathogens. So you can actually, mix that up. Again, I would quadruple or sextuple the dosage that they traditionally recommend for KNF. So I would do maybe a dose per 250 gallons or 300 gallons. 1 gallon per 300 gallons instead of a 1 to 1,000 or 1 to 2,000 like you traditionally. And if you're trying to eliminate a pathogen, if you're trying to treat pathogenic or potentially pathogenic scenarios, it's not expensive at all, and you can treat your whole fields. You can treat acres and acres for less than $100. It's ludicrous. And I was trying to explain this to a farmer over the weekend, and he started off and he told me I was a stupid hippie from California. I didn't know what the hell I was talking about and all this other stuff. And after about an hour of explaining it to him and going through the science and him cursing me out a few times and him telling me I'm a fucking idiot and all this other stuff, by the end of that conversation, he goes, okay, so I'm going to try this on 20 acres of my property. Well, what do I need to do for that? You know, all of a sudden he went from being completely combative and taking it as an insult. And once he realized that I wasn't insulting the way that he does things, I was simply trying to tell him, educate him, and not sell him something. But he's so used to being sold stuff. He treated me like a salesman. And not a salesman. I just wanted to teach him how to do something. I even offered to give him a culture of labs for him to do it himself at his place. And he just couldn't comprehend that because he had not experienced that. So it was so funny to see someone who, you know, told me I didn't know a damn thing. And then here I am an hour later after I argued with, you know, not argue with him, but I sat there and was patient with him and he was being all belligerent. And finally he's willing to try it on 20 acres of his farm. You know, it was real funny to come around.
I have to ask you, when he said, so how do you make it? Did he say, did you hand him your YouTube card and say, go follow my YouTube channel? Because, hey, I just did a video, right?
I'll tell you what I did.
Because Steve just did a video, you guys. Everybody here, he just threw out videos, so check it out. Here he is with his labs.
Here's the labs I just made. Well, actually, I didn't finish doing the follow-up video. I could have done it on Saturday night. It was ready on Saturday night. I've been super busy with the flooding.
Okay, well, we got the first video up. Okay, so there it is. If you need to learn how to make it, Steve's showing you a cheap, economical way. And you know, that is so true. How many people do you approach and you just want to help them, but they're so worried you're trying to sell something because everybody is so used to it. I get it. I mean, I totally understand that. It's not like in our business. I know.
All those labs was I went and bought a gallon of milk and I went to the drugstore next door. And I bought this probiotic. I have no idea what the hell it is, but it has, I'll show you, it has lactobacillus acetophagus in it. And that does a really good job of making large amounts of lactobacillic acid, which you can use, you know, to increase the fungal growth in your soil. And not only that, it loves to feed on the same pathogenic fungi that all the other lactobacillus do. And again, this is like bare bones. I live in the ghetto. I have access to like a CVS or an Eckerd or a Rite Aid, and I can buy a gallon of milk there, and this is all I got. I can't. It's so contaminated. I live in the ghetto. I live in the cities, Philadelphia or New York or Boston or Baltimore, and I can't put out a thing of rice to collect that because I'm gonna get all kinds of car exhaust and all kinds of other crazy bullshit in there. You can go to the store and just buy this, you know, or any other lactobacillic probiotic and milk, and I show you in the video. What did I do? I took the cap off and I poured the whole bottle into the thing in the gallon of milk, and that's it. No sugar, no nothing. Just the bottle and milk. That's as cheap as it gets. It's bare bones and basic as it gets. And not only that, we had stuff within 48 hours. I had ready to use lactobacillic acid.
So was that bottle? Is it one of the refrigerated ones on the refrigerator section? Yes.
Yep. They're always going to be almost all your lactobacillus when you buy it. For making labs, if you're going to do it kind of the ghetto, I call it the ghetto way, or the I don't have access to really hippie friends way, or maybe I live in the middle of nowhere, I don't want people to know what I'm doing kind of way. You buy the probiotic, you tell me you took antibiotics recently, you want to get some probiotic, and it's lactobacillus in it, they'll sell it to you, they won't know none the wiser. You go home, you make it, you're good for your garden.
That's awesome. Master Chip fell out of bed. You know,
so because I have seen people say that they've seen probiotics on the store just like not refrigerated. To me, that just seems like they're getting sold on that, or I don't know.
So you can buy powdered freeze-dried lactobacillus cultures that actually can be used and introduced to milk, but they have a much longer replication time. So you could use that as your initial seed culture, but it would not be good to use every time because it takes you know, a while to wake up and then to replicate versus you know, just replicating a culture. So you can use that the first time, but you'd always want to culture the live culture you know, every time to actually get a better, you know, long-term production.
Okay.
But my frustration is that, you know, with regular lactobacillus, it takes too damn long. I don't have 2 weeks. When I have a problem that I need labs, I need to treat it now. So with this method, I can make a batch in 48 hours. That's actually within the window of time that I could react to something. I should have it on hand, but if I don't, I could actually make a gallon of it, which would treat a really large amount of water, you know, a large area. Within 48 hours, it's an emergency oh shit button. And in commercial, I need that kind of tool. I need something I can whip up quickly and use now. I don't have time to wait 2 weeks. If I'm doing a long-term general maintenance method, absolutely, I'm going to do the traditional method. I'm going to get yeasts. I'm going to get a better spectrum of lactobacilli. Absolutely. I wouldn't. There's no disagreement with me there. If I'm doing, for a regular maintenance dosage for my soil health, absolutely, I'm going to use the traditional method. But if I have a problem, I need to treat something now, I don't have time for that. So this is kind of a solution for a different kind of problem and different kind of scenario than maybe you traditionally use, or maybe you don't have access to the resources that a lot of us do, and this is what you have access to. Or you're trying to stay under the radar and you're afraid about where you're going to get mailed out to your house. This doesn't ring any no one's going to be suspicious about this.
I got another thing about that is that, for instance, they tell you when you put it out, you need to put it in a cool, dry area, even though you're collecting bacteria. And when it's 100 degrees every day, where the hell are you going to find a cool area? But you could do that method you're talking about when you're stressed by heat, Or do you have another idea?
I just put that on the counter. That was literally on the counter of the kitchen behind me. That's it. I got 2 more gallons in there. I'm gonna do a video where I separate it, and we're gonna make 2 gallons out of that 1 gallon. And we're gonna do a video on it just so that you can. And when we get the next batch, we're actually gonna make 5 gallons when we actually get ready to go in the main facility. But until I need that much, I'm not gonna make that video. But we're gonna go through all I have a whole bunch of bag of eggshells. We're going to do the WCA. We're going to go through all the different aquaponic relevant KNF methods. We're going to do how-to videos on it, and I'm going to cover traditional stuff, and then I'm going to cover what I consider kind of the modern, updated way, or ghetto way, or hey, I don't have time for that shit way. How to make this stuff a lot faster, because a lot of this stuff takes too damn long. I have a lot of respect for Master Cho, and a lot of respect for the rabbit holes he introduced to everyone. I think that his methods can be perfected. Or at the very least, when I don't have time for it because I'm trying to treat something, there's certain ways that you can increase production of certain microbes that are the same realm that he's talking about, or but just in a different way that works quicker for my timetable. And I think that's
I love it, because I'm always like, oh shit, I should have done it weeks ago. Shit.
Yeah. I mean, I ain't gonna invent nothing for OHN, but I can tell you for labs, I've sped the process up a hell of a lot.
Yeah. Oh, wow.
See, I've been collecting my eggshells too, and plan on doing a little video as well, because I've done a video on the labs, but I had my labs because it was too hot. I had them, they turned on me. So that my first attempt, and I've been too busy. I'm kind of in that situation where I should have made them 2 weeks ago. Now I need them, and now I don't have any. So, I gotta get off my butt, because like Tara and I were showing off our toys, and I went out and bought this to make more labs, and I haven't even used it yet. So I got to get out there and do it. But that's cool. That's cool, Steve. I was thinking about I saw something on pressure cooking. Somebody was pressure cooking something the other day, and I said, I still haven't seen Steve's thing on pressure cooking. What was it? The coconut oil. Yeah, the coconut oil.
So do you
the
have
traditional sous vide way you were pressure cooking it, speeding it up.
Did you have an update on Dutch blooms and your aquaponics class and that?
Yeah, I was just going to talk about that. So before I get to the class that Josh and I are teaching, Josh is going to actually be teaching a regenerative cannabis summertime home grow session with Shango Los over at Vashon Island Grange Hall. More information at vimea.org or facebook.com/vimea. For more information on that, you'll have a bunch of different seed giveaways and other cool giveaways, as well as a really cool group of people to go talk to. Free parking. It's a free event, so if you're looking to check that out, that'll be on Thursday. Also on Thursday on the podcast, we will have Mike West on the show. So if you're interested about a really high-end extraction, or cannabinoids or terpenes or anything to do with that, someone is extremely knowledgeable. Someone, he knows a hell of a lot more than anyone else I've ever met on cannabinoid expression in particular, and a whole bunch of other cool topics that are we touched on tonight. He'll be on the show. And then Josh and I from Dutch Blooms will be on teaching a 4-day class, Thursday, Friday, Saturday, Sunday of next week at his farm, a commercial aquaponic cannabis farm, where we're going to build a commercial aquaponics farm system at his farm. He also is a DEMPure certified soil farm. So we'll get a chance to go through all of those methodologies and get a chance to work with over 2,000 plants in person. If you're interested in taking that class, more information at commercialapmjclass.com. It's again a 4-day class. It's intensive. We go through design, cultivation, mineralization, mineral balancing, ppm levels, integrated pest management for aquaponics and organic soil. It's a super in-depth class. You get a chance to work with and touch and experience all different types of cannabis cultivars. We'll get a chance to do extraction on a couple of different types of cultivars as well, so you understand the difference between hash varietals versus flower varietals and how to tell that when you're making some hash, sample hash batches, and a whole bunch of other really cool things that are really help tie the whole class together. So it's going to be a really, really awesome time. Again, more information at commercialapmjclass.com. And that class will be in person only in Bellingham, Washington. We will not be doing an online version of that class at this time. Maybe in the future, but not for this particular session.
Yeah. So anybody, anybody into no-till regenerative with aquaponics, it's a unique situation because it's an aquaponics class. You're going to see a aquaponics system built, but the farm is just so vast with so many different methods incorporated in it, along with K&F inputs, that regardless of which way you want to go organically in the regenerative type.
Uh-oh.
We lost Roger. That's all right.
All right.
But yeah, I kind of talked about what I've been up to. We've been just seed sifting. We have, probably 1,600 plants right now that we're sifting through, trying to find, what we want to keep, what we don't, what's going to go outside, what's going to get cloned, and what isn't. So we have about 200 different cultivars that we're trying to sort through right now. So. And some of the stuff we have good cuts of that we've been growing for a couple of months, and some stuff we're still sorting through, and some stuff we just cracked. So, also excited to be involved in the DEMPure breeding program for the hemp production. That'll be a lot of fun. We're going to set aside a bunch of our area just for that. So it'll be a lot of really awesome.
Oh, cool! How much space you putting for that?
What's that?
How much space you putting up for that?
I got to see how many seeds I get. And then we'll know.
Very cool.
However many seeds that's what we're setting aside.
Okay, so how far apart do you plant that type of stuff? I've don't really know much about it.
So you can do up to 2,250 plants per acre when you're doing it in a large scale for outdoor.
Okay.
For indoor, like in the indoor aquaponics, and 6,000 square feet, we're doing between 2,200 and 3,000 plants, depending on cultivar and ultimate size that we're doing. We have it's almost all grow beds. There's enough room to get through and enough room to move the production around, but we can put between 2,000 and 3,000 plants in that 6,000 square feet. Again, doing smaller, quick turn stuff, not doing large plants on the indoor greenhouse space.
Right. So if someone like me was interested in growing a few of them, could you grow real big ones and just kind of?
Oh yeah, so our outdoor plants will all be multi-pound plants.
Okay.
Yeah, our outdoor will all be single season, and then our indoor, we're not going to do any light depth. We're going to do too many plant. We could do it, but we have a lot of space that we can grow, and we're going to fence in an enormous area. You have to have an 8-foot fence around your cultivation space in Oklahoma, and we have a huge space that we're going to be fencing in. So we're just going to grow basically whatever we're going to keep cloning and taking cuts and growing what we have until mid-July, and then we're going to stop planting outdoor. So that's kind of the game plan.
Very cool. Well, thanks. That's a great episode tonight. Like I said, you're all watching, you go back. Cheers, guys.
Yeah. What about you? What's going on with your garden?
Like I said, I'm probably gonna plant this week. You know, it was 84 degrees today, so I think my babies are wanting to sprout out. The problem is I don't know, you know, if I got male or females yet. So
you could buy one of those genomic testers from him or for that group and test all your stuff right at home.
I'm kind of thinking about it. Like, yeah, maybe I should. How much are they? You know, I want to know some more.
Right?
Yeah.
Wow. So was it me that froze up? I lost you guys for a few minutes. I didn't know what happened.
I can't hear you anymore. What's going on with you in your garden? Anything?
Talking to me?
Yeah.
Oh, well, we kind of addressed that earlier. It's just been 100 degrees out here and trying to keep my plants from melting. Got the shade cloth out. Going to put up some. I got a whole bunch of pipe. I got top rail pipe I got from the lake, from the military complex a few years ago. They're like 18, 22 feet long metal poles. And I just cut them to the length I want, beat them in the ground, and I can hang things over them like shade cloth or plastic or whatever like that. So I'm going to try to get out there and not die from heat exhaustion tomorrow and get some of that done and make a space for my trees and stuff. You know, that's basically what I'm doing. Just trying to catch up. But we had, like I said, it snuck up on us. It was so mild, and you know, it was maybe 85 to 90. Then all of a sudden we got this weather. So other than that, no, I'm sorry. I wasn't sure who blanked out, and I tried to get back 2 or 3 times. So, you know.
So DK, so worried about what size they are, or what sex they are. And so I think I am probably gonna, once I'm still worried about what sex they are, I'm gonna probably throw in bigger pots. And then the clones that I have that I've gotten for my friends, I will throw them into my grow beds. So I'll at least have those. So that's what I'm doing. Did we lose Pony?
Are you sexing them to for cloning and to keep mother plants?
No, not yet. I just planted seeds, and whatever I get, that's what I'll be growing this year.
Oh, so you're gonna?
I'll give them away.
You're gonna call the males basically then, and keep.
Yeah, I'm just gonna get rid of them.
Old school. Okay.
I'm doing breeding. I'm not a breeder. I don't.
Are you going any more of that orange cleaner?
Oh, there you are. Oh, the orange cleaner. I have a friend that told me he's got 3 seeds for me.
Yeah, I think everybody wants that.
You think?
She had a super amazing orange cultivar that was really wonderful.
Yeah. It was an orange cleaner. I know, right? So I was watching the 360 movie or the video idea of us walking into the 420 event, and we sit right down on the hill and we have our first dab right there, and you're like, serious orange cleaner. It was so funny. I was like, yep. I got to get that video uploaded.
Alrighty, well, I think we'll wrap things up here. How does everybody find you, Roger?
Sorry, you can find me at I Love Growing Marijuana. Excuse me, hold on.
You took a nap.
How's everybody find you, Tara, while we're waiting on Roger to stop choking?
You can find me at Tara Lee Live now on YouTube and on Instagram.
Sweet.
You can find me on Instagram and Facebook under Roger Latewood. Really, my whole something we never. Well, it was nice. Steve was trying to, I think, keep me incognito throughout the first. Just because of where I live and all, and I appreciate that. But I've always been known as Latewood in the cannabis industry for over 15 years. And so since everybody knew me as Roger, I went ahead and adopted Roger Latewood this year. And I'm at Instagram and Facebook. And I did have a couple people contact me on Facebook this week, which is nice because the PM. And I did have a friend, a shout out to Sid. I got to get with Sid later because he came up with I'm always bitching about Instagram, how it's not made for computers, it's for phones. And he found an app that I can download to my computer, and I'll be able to use Instagram like people with a phone. So I'll be able to message everybody, all my friends I met in Michigan. I said, yeah, I still can't believe I hadn't really used Instagram much when I told everybody, oh, I'm just going to start an Instagram account, and then I found out there were some problems with that when you use a PC. So, but Sid, one of our buddies from Good Earth, he's always digging stuff up and trying to help people. And he came, he was in a rush the other night. He told me what it was, and before I got exactly what it was, so I could go get it, he had to go. So hopefully I'll talk to him tonight or Thursday night after the show, and I'll be able to hit you guys up on Instagram, and message you and stuff like that. So looking forward to that.
Very cool. Awesome, Roger. How do they find you?
So you can find me on YouTube at Ponics, SoundCloud, iTunes, Spotify. We're also now live broadcasting on twitch.tv. That's why you guys can see the stop broadcast button, why we don't have a private chat anymore. I'm going to work on cleaning this up and making it nicer. I'm still getting used to the whole OBS thing. I actually streamed this whole episode via a cell phone. So please bear with me while we're getting, I don't have internet in my name yet out here. We're working on that. So until I have a cable line, we're going to have a little bit of some technical problems, very minor ones, going forward. But you can find the recorded version on the podcast app of your choosing. I have it on everything I can get my grubby little hands on. So thanks for watching. Big shout out to Kevin McKernan, medicinalgenomics.com if you want to find out more information on him or you want to use his services. Thanks a lot for clearing the airwaves, and much love. Thanks a lot, and take care. We'll see you guys again on Thursday with Mike West of Embark.
Yes, Cheers. Mike West of Embark. Cheers.